I think it does. Okay, so good afternoon to everybody. We will start spot on because we have a very rich program. And I asked James, who just becoming the head of the department with this challenging role, so he kindly agreed to give some welcoming notes to our masterclass symposium. So James.
Thanks very much, Latsi, and welcome everybody. It's really exciting to welcome you to this online Masterclass Symposium. I was just looking through the program. Latsi a moment ago said that it was challenging. It certainly is. It's really exciting just to read the titles of your talks and your posters and the number of different schools who are involved. And the scope of this project, this whole outreach project that Latsi began a few years ago and has now really expanded. COVID has, produced all sorts of challenges. It's been a dreadful time, but one of the few silver linings, I suppose, from the pandemic is that it's become a bit easier to organise this sort of a event, bringing together staff and students from multiple institutions, from the comfort of our own homes or wherever we are. So I suppose we should be grateful for those small mercies. But really looking at all of these topics and the depth of the science that you've all been engaging in over the last months. It's I can tell is going to be at a very high level. So I'm really looking forward to seeing your posters and hearing your talks. And I'm going to be seeing you in about an hour's time for a bit of an online practical session at 5 o'clock. But before we get there, we've got lots to do in the first couple of sessions. And then I'm going to hand over now to Dr. Deborah Harvey, and Deborah's going to say a little bit about studying biology here at Royal Holloway. So I know I haven't quite used my 5 minutes, but I'm sure we'll benefit from the time. Deborah, if I may hand over to you.
Thanks very much, James. So yes, my name is Deborah Harvey, and I'm going to talk to you this afternoon about, I'll get my slides to share. Oh. Sorry about this.
We start to see it now.
About
Fine.
About studying biological, lovely, studying biological sciences at the Royal Holloway. We're hoping that some of you, after today, some of you will love to join us at the Royal Holloway. But why would you? Well, at biological sciences at Royal Holloway, we're one of the top 25 UK biological sciences department in this country. So you'll be joining a really good student community taught by really good, respected scientists. But it's more than that. It's about you. And what we can tell you that if you come to the Royal Holloway and study biology with us, you will contribute whilst you're doing your third year project to real research. And for those of you that, and I'm sure it's all of you here, that enjoy practical biology, our degree is based on a large amount of practical teaching, and unusually, we give you choice from the very first year of your degree. So when you finish your degree here at Royal Holloway, you've got your own degree, not just a biology degree. But is it just what we say? No, our students love us too. So we've got really high overall student satisfaction across all of our degrees, and I'll tell you which ones we have in a moment. We had 100% overall satisfaction from our students in 2020, and we're 7th in the UK for graduate prospects. But more than that, we've got a really good, strong record for graduate employability. And at the end of this presentation, I'm going to tell you some of the places that our graduates go. But 96% of our graduates are employed or in further education within 6 months. We really think that you're important here at the Royal Holloway. So we have very supportive learning environment. As well as the department, we also have a department which is called CDAS, which helps you if you're struggling with to write essays, if you are not sure or you need a bit of help with stats or whatever. And in the department, when you come to us, you'll be given a personal tutor that is one of the academics. You and a group of another, say, 6 students will spend the next 3 years working and spending time and talking to that academic who will be your personal tutor to talk to you about anything you need to talk about, whether it's writing a reference or your CV or anything at all. At the bottom, you'll see I've written that we've got Royal Society of Biology accreditation for all of our degrees. Well, so why is that important? Well, the Royal Society of Biology only accredits programs that are really good. So this shows your degree is really excellent, which will give you an edge when you apply for a job, because this is such a crowded job market now that you want everything to help you, and this will help you. Because why? Because it tells industry that you have the employability skills and the subject relevant skills to make you a very good employee. So what are these biology degrees I'm talking about? Well, I'm going to say we have 8, but we have more than that. We have medical biochemistry, and biomedical sciences. For those of you that are really interested in medical biology, genetics, and then we go more to the organismal side. So we've got biology. And sometimes people say just a biology degree. It's not just a biology degree. It means that you will have a wide, broad-reaching degree that covers all areas on this slide. Zoology, for those of you that are interested in animals. Ecology and conservation, for those of you that are becoming really aware that we need to do something to help the planet and save the species on it. And our new degree, molecular biology, environmental change. I said to you there are more than 8 different degrees, and that's because each of these degrees you can have a year in industry. So when you look at UCAS, you can see that you can apply to have a year in industry. And what does that mean? Well, it doesn't just mean you can go and work in industry. It's a blanket term for you being able to take a year out of your degree and to go and work maybe in a company or with a charity or whatever you would like to do to work. Supposing you don't do that, is that the end of your options? No, because in our second year, you can also go for a competitive scheme to take a year out to go abroad and study something that you're interested in. So I said you can choose many options from the first year of the course. And in the first year, you do 8 15 credit modules. Now, depending on which degree stream you choose, there'll be some that will be core and you'll have to do. But if you look here at the range, and you'll see these slides later, they'll be available to you of the subjects that you can study. So you might want to do chemistry, you might want to do animal physiology, look at the effect of humans on the world today in biology in a changing world, or study plants, green world, plant evolution, form and function. Moving on to the second year, and you can see it doesn't change, it gets even more diverse. So now we've got even more units, many more than we can show you on this slide, that you can choose in your degree stream, whether you're interested in behaviour, pharmacology and toxicology, insects, plants and fungi, or developmental biology and plant biology. There are so many options. Moving on to the third year, you'll see again, and I'm not going to read these to you, but there are a few here: circadian biology, population and community ecology, marine biology. Really so many options, gives you such a wide choice to get your degree that you want. I said to you at the beginning, we're very hot on concept on biology and field work here, and so we are. So we have our own practical field ecology course that we hold on campus and in the local area. And why wouldn't we? We've got, as well as the beautiful Founders Building, we've got wonderful grounds here. Hopefully sometimes you'll be able to come and see these. And there are so many wonderful species here, but as well as looking at the species on campus, we've got heathland around in this area, we've got Staines Moor, and we've got, of course, Windsor Great Park. But supposing you fancy going further afield, well, we have a field course in Scotland, in Millport, a marine biology course. Even though we're not near the coast, we still do marine biology here. And Professor Dave Morritt, who's an expert in plastics as well as many other species of marine life, will take you to Scotland to investigate marine biology. For those of you that fancy the Mediterranean, you can go on a Portugal trip where you'll be, as you can see in the picture below, doing some diving, or to go to the rainforest to learn about the Borneo rainforest, to learn about the indigenous tribes there and the biodiversity you find there. In the third year of your degree, all of you would do a research project. Now, this is for over 2 terms and is a whole unit. So 2 of those 8 units you do in modules you do in a year. What do you do for it? Whatever you like. You have a wide range of topics on virtually any aspect of the biological sciences. And you can either follow one of those projects or your own interests and develop your own project. And many of these projects will lead you to think you might want to do a master's or a postgraduate study, and may even become part of a publication. And here you're going to work as a scientist in a lab led by an academic with the academic team. The research in this department is very strong. 76% of the research was rated 4-star or 3-star. And you can look here just to see the range, and this is just some of the areas that we research in this department. So you'd be hard pushed not to find something that you liked. What's a typical day for biology undergrad? Well, in your first year, you'll have 4 days a week with us, working 9 till 5. So the morning typically will be lectures and the afternoon will be practicals. The second year, we go down to 3 or 4 days a week, 9 till 5 again, same practicals and lectures. The final year, you don't have practical, but don't be misled. That doesn't mean you're spending all of your time in the lecture theatre. This means you're doing your final year project and you're spending a lot of time doing your own research. For Wednesday afternoon, you can join all of the undergraduates and postgraduates across the university, having fun and engaging in extracurricular activities. Where do our students work? Well, a lot of them will stay on to do master's degrees and PhDs. They'll go on to do medicine, dentistry, veterinary science. They'll go on to teach. But here are just a few of the other places that our graduates have gone over the last few years. And you can see there's a very, very wide range of places that our graduates, 96% of our graduates, remember, are employed or in further education within 6 months of graduating. And there's a very, very wide range. When you get a good biology degree, you can go wherever you like. Thank you very much for letting me talk to you today about our courses at the Royal Holloway, and I hope you enjoy the rest of this afternoon. I'm sure you will.
Thanks a lot, Deb. And so we will push on and get to our main program. So let me share now my screen. And so I am really very grateful for the fantastic contribution and the active participation of our students. So we have such a lot of nice talks and posters assembled. So just few words about the housekeeping. So we go with an hour program of talk session and poster session. So the talks I will be sharing the slides, and the students, everybody should have an enabled that they can unmute themselves and speak. So please, when you are not talking, mute yourself back. And then the posters, I will be quickly flashing them up, but I am expecting that you will go through them also within your own time. And they are all up on our Padlet. And so how it looks is that these are the posters, and I am hoping that you will be commenting on the posters as well on the corners. What I also set up, which is in front of the block, that you can score each other's posters. So this is a scoring sheet, and we will be awarding prizes, both the scores of the participants as well as I will score them as an academic together with other academics who are present. So please use this scoring sheet about the talks and the posters. We have separately for the posters, and the same I put up the talks, and in front of the talks there will be a scoring sheet for the talks as well. So that we are not going just talks, posters. So I broke up the program with 3 practicals, which I think is very exciting and reflecting that we think that biology is not a passive subject. It is not a cookbook, and we never get to be cooking. So the important thing is that we do experiments and do uh, research and get used to this research mind. So these 3 practicals will be one about genetic engineering on a topic which is James set up this production of tissue plasminogen activator. Then the second is that we do want you to like plants, as you already recognized from the talks as well. So Enrique modify the practical which he is running in the second year, that it can be usable also for A-level students. So he will present this idea, and we do want to these ideas of practical work to be adopted to schools. So I am very happy to discuss this later with the teachers as well. And then the third practical part is Ali just came back on this Friday from one of the field course James just mentioning. So he made a little uh, presentation and actually recorded it as well about this field trip. So Ali will come on at around 7. So I hope we get through everything in time. And let me now start and introduce our first speakers, which will be Jasmine and Leah, and they are talking about a modified DNA called zDNA, a very exciting and unusual form of DNA. So I am sharing the slides, and Jasmine, Leah, and Tiffin, are you on the?
Yeah.
Okay. So then the floor is yours.
Okay, so ZTCG, viruses expand the genetic alphabet, by Jasmine and Leah Tiffin. Next slide, please. So the basics of DNA. DNA is composed of molecules called nucleotides. Individual nucleotides are made up of 3 components: a phosphate group, a pentose sugar, and a nitrogen-containing organic base. The 4 types of bases are adenine, thymine, guanine, and cytosine. Adenine is complementary to thymine, and guanine is complementary to cytosine. The ratio of adenine and thymine to guanine and cytosine varies from species to species. The pentose sugar, phosphate group, and organic bases are joined as a result of a condensation reaction to form a single nucleotide known as a mononucleotide. The continual linking of mononucleotides by condensation reactions forms a long chain known as a polynucleotide. Next slide, please.
Can you see the next slide? So we might have lost that student. Ah, so it would need to be unmuted. Am I muted as well? So because now we can only run by allowing everybody to mute and unmute. So it's very important that our speakers are. So I can see Jasmine is muted.
Leah's microphone isn't working. It won't let her unmute.
Ah, okay. So can you present then Leah's part?
I think she had other stuff to say, and I don't know the extra stuff that she had to say. Sorry.
Okay. So then we go through this slide and the next speaker, but essentially it is the history of discovery of the zDNA, which at those time was simply about the base pairing with the thermostability of the DNA. So heating it up and separating the bands will tell how strong is the pairing between the strands, and that showed that this is unusually strong in this modified base. I think that is on this slide. And the next one then, so do you have the next student?
Yeah, so DZTP and DATP. So DZTP, otherwise known as 2-amino 2-deoxyadenosine 5-triphosphate, is a putative DNA polymerase substrate, and DATP, deoxyadenosine triphosphate, is a purine nucleoside triphosphate used in cells as DNA polymerase substrate. Recently, the Institute has discovered a type of phage polymerase that incorporates DZTP into DNA whilst removing any A bases present. This explains why A bases are excluded in the form of DATP. Another research team believes that additional phage enzymes are required to break up DATP whilst preserving DZTP inside cells. They discovered that increasing DZTP levels relative to those of DATP is enough to trick a cell's own polymerase into creating zDNA. Next slide, please. So the uses of zDNA. zDNA, located after the promoter region, is thought to increase the rate of transcription, with the greatest increase in activity observed when zDNA sequence is placed 3 helical turns after the promoter sequence, which means mRNA can be synthesized faster. mRNA can be used in many different specialized treatments. For example, in the Moderna and Pfizer BioNTech vaccines for the COVID-19 virus. Cancer treatments are also being developed using this same technology. This is because you can easily encode the desired proteins. zDNA forms 3 hydrogen bonds between the Z and T nucleotides. This would have provided an advantage to early life forms with this Z genome, as this prevented the hot conditions of the early planet from unzipping the double strand of its DNA. Now, this can be utilized in DNA origami, as the bonds between the branches of DNA are stronger, so the structures developed will be more robust and less resistant to being broken down. Next slide, please. Thank you.
Okay, that was great. So any burning questions, please? Until anybody has a question, I have one. So you talked about this interesting topic of DNA origami. So do you know what that is?
Leah did that section, so I don't know whether she can type it in the chat.
Okay, if she could, yes, but it is essentially that the DNA is a fantastic polymer which can be used to fold in any shape and form. So, so they can, for instance, make a little basket, a miniature basket, which can go within cells and deliver a drug and things like that. So it is about this folding. Any further questions? If not, then we go to the next talk. Thanks a lot, guys. And you can keep submitting your questions at the corner on the Padlet or in the chat box. But now we go to the next presentation, which is about gene editing and specifically curing sickle cell disease. And this will be by Alezach. So are you around? Can you unmute yourself? And we also have two more students, so Sophie and Azar. Oh, the sound is not really great. Yeah, the sound unfortunately is not very good. That's the problem with the online challenges. We need a good connection. And I don't know if the others hear it. I can't, I only just hear noises. But what we can do if we can't solve this quickly, that I can go on to the next talk and we come back to this. If that is okay, because we won't be able to listen. We come back to this, and so let me go to the third talk, which is an interesting topic of this cardiomyopathy and essentially printing the tissues of heart. It's a fantastic new technique. So if Hunda and the group is around, are you around?
Hello, can you hear
Hello.
me?
Yes, we
Hello.
can.
Hello. Okay, so I'm Harshini and I will be talking you through the presentation on Takotsubo cardiomyopathy. So what is Takotsubo cardiomyopathy? Well, it's more commonly known as broken heart syndrome, and it's a real condition that exists outside the work of television. So it's essentially a disease triggered by extreme emotional distress, which causes the inflation of the left ventricle. This distinct shape caused it to be named after the Japanese octopus trap due to the inflated shape of the end systolic ventricle. Takotsubo was first reported by a Japanese cardiovascular specialist in the 1990s. It's mildly independent with the acute myocardial infarction, also known as a heart attack. So, unlike a heart attack, which is commonly caused due to blockage of the coronary artery, there's actually no evidence of blockages in an angiogram. So, another key marker of this disease is the rapid but small rise in cardiac biomarkers, which in a heart attack would take longer to rise and would peak higher. However, similar presentations make it a confusing diagnosis, as patients often complain of severe chest pain due to the distinguishing factor being the lack of blockage. Triggers of the diseases are factors that cause a drop in blood pressure, receiving bad news, the loss of a loved one, serious illness, or a medical procedure, such as cardiac stress or a domestic violence. However, the exact cause of the disease remains unknown. Scientists suspect the reason is elevated levels of stress-related hormones, like adrenaline, which trigger changes to the cardiac muscles. To test the impact of mental health on Takotsubo, researchers made 2 comparison groups. So they had the healthy controls and patients with chronic heart failure. Both groups of men and women took standardized questionnaires, and they took into consideration things like depression, perceived stress, anxiety, and personality factors. And the results obtained that patients with Takotsubo had a higher level of depression, stress, and general anxiety. In terms of genetics, an investigation was carried out to determine whether genes play a role in Takotsubo cardiomyopathy due to the aggregation of the disease in sisters or mother and daughter. Specifically, the study was to investigate the possible associations between Takotsubo and a single nucleotide polymorphism in ADRB1, GRK5, and BAG3 genes. The method was essentially 3 groups, and they had their Takotsubo patients, controls with coronary artery disease, and controls without coronary artery disease. These groups were age and sex matched, and the DNA was isolated from the saliva and genotyped for the single polynucleotide polymorphisms in the gene specified before. So, and then they filled out questionnaires as well, answering things like emotional and physical stress. And they had their mean, standard, and deviation tests, and they found that the allele frequencies did not differ between the 3 study groups and did not differ between men and women. So they were consistent with the reference populations in the 1,000 genomes database. However, scientists still think there's a link between genetics and Takotsubo, and more genome-wide approach needs to be done. In terms of relationship between menopause, a test was done where there was tests conducted on mice, and they were divided into sham operation and ovariosectomized. And sham surgery is essentially a placebo surgery, and which is a faked surgical intervention that omits the step to therapeutically making it therapeutically necessary. So 6 weeks after the ovariorectomy, the plasma levels of EPI, also known as adrenaline, significantly increased. Then they were injected with EPI to make stress-induced cardiomyopathy models. Lack of estrogen resulted in the EPI surge and more serious cardiac dysfunction and higher cardiac troponin. So estrogen deficiency also decreased the concentration of CAMP and pKa in OVX plus EPI group. So in turn, researchers have concluded that the lack of estrogen replacement in menopause causes women to be at a slightly higher risk of acquiring Takotsubo. However, more studies need to be conducted to establish more strong links. Next slide, please. In terms of recurrence and treatment, Takotsubo can't be treated itself, but the symptoms and the structure of the heart can be. So diuretics, I've included a picture here. They essentially help to stop fluid buildup and reduce it. Beta blockers and ACE inhibitors take the strain of your heart and help it work more efficiently. And blood thinning medication also reduce the chance of blood clots. Recurrence is clinicians should be aware that patients can have recurrence episodes. So there was a woman that came in. She was a 76-year-old woman, and she had a total of 6 recurrent Takotsubo episodes spanning over 33 years. And her diagnosis was first made in 2014 when she presented with chest pain, raised cardiac enzymes, and the presence of normal coronary arteries. Cardiac magnetic resonance was performed, ruling out any previous myocardial infarction. Subsequently, she had 2 further recurrences in 2015 and 2018, and stresses were identified on all 3 occasions. She was also diagnosed with myocardial infarction in 1986, 1988, and 1998. Because they didn't know a lot about the Takotsubo in that time, they ruled it off as just a heart attack. But now that they've done a little bit more research on it, they can figure out that it could have possibly more likely been Takotsubo. There is a picture of an ECG over there of a patient who did have Takotsubo. It's not the 76-year-old woman, it's just a normal patient. And you can distinguish it from a normal ECG, there's a ST segment elevation. It's not as clear maybe, but there's also a ST segment depression, and there's also a left bundle branch block and a prolonged QT interval to help you distinguish it from a normal ECG. Yeah, thank you for listening.
Thank you so much. Any quick questions? I need to go back to my chat box if anything burning happening there. But if not, then I think we also just push on. It was a great presentation. Thank you so much. I had a not so good introduction to it, but the topic was nicely described. So the next one is the mechanisms of synthesia, and this is from Herschel, Gramme, and Mariam and Seema. So are you there?
Yes, sir.
That's great.
Mariam, can you hear me? Yeah. Okay.
Yes. So then the floor is yours, or the screen.
Welcome to our presentation. My name is Seema. And my name is Mariam, and we'll be talking to you about a condition called synesthesia. Synesthesia is a trait which around 3% of the population have, and it's where a person can perceive information through more than one type of sense. For example, associating both colours and sounds for one thing. A way to understand this is using the concept of metaphors. You can say that synesthesia is a condition in which we will perceive metaphors as senses. For example, when listening to a song, a person with synesthesia will associate a colour or taste to the song as they experience it, not just like as a sound. Next slide. Yeah, it's on slide. The mechanism of synesthesia. Graphemes is a type of synesthesia which exclusively associates colours and letters and numerals. Similarly, phonemes associate sounds with colours, interlinking other sets of senses to create a process of information that's different to normal people. Many artists in different fields, such as music and literature, are known to be synesthetes, and they have gone on record to talk about how this condition has had a great impact on the work they produce. The perceptions they have due to the mixing of the senses provide them with inspiration, and that allows them to create their art. Next slide, please. So before research had been done into this condition, people who shared their experiences with synesthesia were labelled as crazy and were thought to be abusing drugs due to how odd their experiences with their senses were. It took a lot of research in order to clear up this misunderstanding and to legitimise synesthesia as a real condition. George Sachs wrote a dissertation about albinos, and in that dissertation it contains early research on synesthesia. Directly quoting his dissertation, he said, "Simple series, for example, numbers and the days of the week and such similar things, adopt those colours." His research implied that he noticed people who experience information using more than one or non-corresponding senses. This example was seen as the first scientific study detailing synesthesia and the symptoms of the condition. After the initial research into synesthesia, the fascination for the topic plateaued and was forgotten for a long while until Robert E. Cytowick reignited the discussion in the 1980s and returned the topic back to mainstream science. Robert E. Cytowick ended up making many breakthroughs in the topic, pushing forward further research into the phenomenon. He also published many books on the topic, including his award-winning book simply titled Synesthesia, as you can see on the slide. As well as other books. Next slide, please. So thanks to Cytowick and the interest he had attracted towards synesthesia, we were able to use modern technologies to garner evidence for the condition and gain a better understanding of it. Using the MRI scans and looking at the parts of the brain that were stimulated when a piece of information was perceived, researchers were able to observe different sensory pathways lighting up, which would not match the sensation being experienced. PET scans were also used to look into synesthesia, and it was found that people with the condition may experience greater cortical deactivation. For example, when you see something, your visual cortex may be deactivated, so you will not perceive the information as vision at all, and you may perceive it as a taste or as a sound instead. Next slide, please. Research found that synesthetes have a denser grey and white matter, which the brain is made up of, providing supporting evidence that grey and white matter are responsible for processing information. The electrical impulse enters the grey matter, which then is processed, which processes the information and directs it into the white matter, which is made up of myelinated axons. These will construct a tract, like a pathway, sending the impulse to its corresponding locations. Grey matter can be thought of as like a gate in the brain, and through that you will access the white matter, which will continue the path to the spine and stimulate a response.
At this point, the impulse may jump between the tracts, allowing the synesthetes to perceive the information through different senses. What was particularly interesting about synesthesia is how the perception of this condition has changed over time due to research.
People who were once labelled drug addicts and were thought to be mentally ill are now thought to be special if they possessed a rare synesthesia. Next slide. Thank you for listening.
Yeah, it was a fantastic presentation. I can't clap enough from here. It was very nice. Any questions? If no questions, we can have an effort to go back if the sound would improve on the second talk. Can you unmute yourself, gene editing group? We still have the same so I think my narration would be a very... Hello?
Still unclear.
Now it's better. Yes, so
Okay.
let's try.
Try now. Okay, good afternoon. We'll be presenting on CRISPR gene editing to cure sickle cell disease. Next slide, please. To begin with, what is sickle cell anemia? It's an inherited disorder caused by a point mutation in the gene which encodes the beta-globin chain of hemoglobin. The mutation results in the replacement of negatively charged glutamic acid by a hydrophobic valine. This results in the distortion of the red blood cells, and this can cause blockage of circulation, resulting in acute severe pain episodes called sickle cell crisis. Management and treatment of the disease is usually aimed at avoiding pain episodes, relieving symptoms, and preventing complications. The only treatment that offered a potential cure is stem cell therapy. Unfortunately, this isn't available for all patients because it can be difficult to find a suitable donor. Next slide, please. Clustered regularly interspaced short palindromic repeats, otherwise known as CRISPRs, are repeating DNA sequences in the genomes of prokaryotes such as bacteria, which was first identified in E. coli in 1987 by Japanese molecular biologist Yoshizumi Ishino. The lab-created system consists of two key molecules that introduce a mutation into the DNA. These are an enzyme called Cas9, which acts as a pair of molecular scissors that can cut two strands of DNA at a specific location in the genome so that bits of DNA can be added or removed, and a synthetic piece of RNA called guide RNA, which is designed to find and bind to a specific sequence in the DNA. Cas9 follows the guide RNA to the same location in the DNA sequence and makes a cut across both strands of DNA, and so far it has been this system which has been the most notable in treating sickle cell disease. Next slide, please. CRISPR as a treatment was used to genetically modify a patient's bone marrow stem cells to produce high levels of fetal hemoglobin in their red blood cells. These are then delivered back to the patient as part of a stem cell transplant. It was in mid-2019 when the first patient was announced to be enrolled in the study, a 33-year-old woman who had experienced 7 vaso-occlusive crises per year. 4 months after the treatment, her fetal hemoglobin levels had increased from 9.1% to 46.6%, and the percentage of fetal hemoglobin producing red cells had increased from 33.9 to 94.7, and there are positive results as shown in the presentation. Next slide, please. Overall, though being a new innovative biotechnology, it also carries its own limitations and setbacks seen as it's a fairly recent discovery. One of these include off-target effects. In theory, CRISPR-Cas9 system should be incredibly specific. However, in practice, it's not always the case. It can create mutations elsewhere in the genome known as off-target modifications. However, these defects are of low risk, and researchers are working towards eliminating them. There are now a variety of fields where CRISPR can be applied to, from healthcare to agriculture, with examples being the production of golden rice and cancer immunotherapy. It has led to many believing that the technology will pave the way for curing many human diseases and even combating climate change. As said by the chair of the Nobel Committee for Chemistry, there is enormous power in this genetic tool which affects us all. It has not only revolutionized basic science, but also resulted in innovative crops and will lead to groundbreaking new medical treatments. Next slide, please.
Which is
Thank you.
big thank you slide. Thank you so much. I am so happy we could give you the chance to present and not falling down with the sound problem. And any questions? That so my note to this. We also have a nice poster about the same topic, so please pick that up as well. And it was a very accurate.
I've got a question, Lati, if I
Yes.
may.
Yeah, sure.
Thanks for the talk, Renuka. Do you have any idea why it is that the red blood cells, why it is that the hemoglobin in sickle cell patients, why does it only stick to other cells? Why does it become sticky in the veins and the capillaries rather than in the arteries? Do you know why that might be?
I'm not sure, but I think it's something to do with the hydrophobic valine being produced, and it's less soluble, so there's like a lower affinity of oxygen.
Yeah, that's definitely true. But it's only on the. It turns out it's only on the surface of the protein when it's deoxygenated. So when hemoglobin is bound to oxygen, it's not really a problem because it's not on the surface. But when the protein changes its shape when it binds oxygen, and then it's on the surface, and then it becomes sticky. But that was a great talk. It's really interesting.
Thank you.
Okay, thank you so much. And so now we will go to the posters, and as I told you, please go on the Padlet and pick up the talks and the posters and vote as well. Don't leave your voting to the very end. As we go on, the best way is to try putting your votes down. And so the posters, so pick them up on the Padlet. And just to know that what posters we have, I will quickly run through them and just give a little teaser of the posters. So let me see if I can open. Okay. So the first one, the poster is about antibiotic resistance from Loretto School, and it is Z. And so essentially this post nicely tells us that this is such a huge problem that the overuse of antibiotics are taking away this essential it is an absolutely essential tool. You can remember back of the surgery lecture of James. And it's also telling us this post that it's not a big business to run after this for the pharmaceutical companies. So there is also a problem where we and you can enter this field and change this field, that medicine is not driven by business, but it is driven by the need. So this is the first poster. Then the next poster is a similar talk on the CRISPR, and and uh, and this is very much in line with the talk what we heard. It is nicely describing the mechanism, how it was discovered, what it is doing, how it is doing, and some of the use, which this is one area which convinces us that to go to biology is absolutely a good choice for you guys, because it's a fast moving train. So if you want to do exciting thing, you want to go with a super fast train, and biology is one. And the gene editing proves that it was just discovered like 10 years ago, the use of it, and it's already becoming into the practice. So this post tells all about this. So then the next one is again going about the antibiotic problem, the cycle of antibiotics, and this post is from Arnold Hill, and it is a similar name, but I believe a different student called Ziyu, and and this one goes a bit more into the details of putting down data of the different antibiotics and the different generations of antibiotics, and how we are running out of options, and then going into the description of what we can discover as a new tool to replace the non-working antibiotics in one hand. The other hand is that how we can solve the problem by not overusing antibiotics, and for instance for animals, and getting into our food chain through agriculture. I hope I am describing it well. And anytime if the student have a comment I forgot to tell, please unmute yourself and chip in. Anything you want to add? Or put it on
We do have one comment from a student, Renuka Rekar, who says that her group had a group presentation as well. But did we miss one off the schedule?
About antibiotics.
Renuka, do you want to unmute and explain?
Hi, Professor. It was
Hi.
about buzz pollination and the decline of bees.
Okay, so I missed it in the program.
Oh, okay.
No, I am asking, but if I missed it, I will give you, because our third session is the ecology related session. So we will give you the floor in the third session. And then best way to do it is that you share your screen when we get to that point. So just before 7.
That's fine, sir.
Would that be okay?
Yeah, that's fine.
That's
Thank
great.
you.
Thank you telling us. I am so sorry that I am making these mistakes. Okay, so the next post is about a disease which is called congenital insensitivity to pain, and this poster very nicely describes both the symptoms, which essentially that these patients are not sensitive to pain, which is a very big deficiency if you think about it hard, and also goes into the mechanism, that it is a mutation in a particular protein which is important for the receptor of the neurons, and it's also sending these neurons to die and losing these neurons. And then here it is describing some of the possible treatments, and one of them is a drug which works completely the opposite, as sensitizing, uh, desensitizing neurons, so the painkillers like opioids, so it will sensitize these neurons, and in that way bring back some of this pain sensing. So this is a nice poster. And then the next one is another disease which is autoimmune disease. It's from Arnold Hill, and it's from Eve and Adele. And this disease is about gluten sensitivity, and that the autoimmune system is activated, the immune system activated against the own, and will start destroying the lining of the gut. And because of that destruction, the absorption of vitamins and also essential elements from the food is compromised, and this will lead to many other problems like diabetes or this thyroid disease, which is a consequence of destruction of the gut system. So that was, and here I would like to note that this is a nice reference from a leading medical journal called Lancet. So this is a good example for fantastic referencing. Any comments to this? If not, then we come to this poster, which comes to the problem of plastic, which is a huge problem, and a scientific solution that bacteria, some of the bacteria can have enzymes which are degrading the plastic, so eating the plastic. And this is an example of enzyme design by looking in silico the genes in different bacteria, and trying to find genes which are having this activity of polyethylene hydrolase that essentially degrading plastic, and very important research. And I believe the last one is about memory. So I also recommend to pick all of these posters up. So this goes back to the lecture, the nice lecture of Philip, which talked about the memory. And the poster provides a nice summary of where the memory is working within the brain, and the discovery through these patients, and the surgery of this patient called HM, which destroyed the hippocampus, was removed and destroyed part of the brain, and led to the discovery that specific parts of the brain is responsible for specific elements of the memory. So these were the posters. Any comments to this? I was running through them to save time, but please put your comments to the posters, and also look through them, and also give the students feedback by voting for the posters with the different numbers which I set up. Okay, so I don't hear anything, so I think we go back now to James to switch out to a different activity, which is a practical. Well, if you would have come to Royal Holloway, you would enjoy it in our laboratory, teaching laboratory with James. Usually when we had it on site, this was a highlight of the day for the students. But James, try to do your best to replicate that experience. So I unshare my screen, and the screen is yours.
Thanks, Latsi. Hopefully you can see my screen. Let's just let's do this. Okay.
Jay made a little video about this as well, which I put up on Padlet.
Oh, thanks. Yeah, I'll show that in a moment. Hello, everybody. So this is the lab that we would have been doing if you could have made it to campus today. And it was all about cloning a gene and using electrophoresis to find out if we'd successfully got that gene into some bacterial cells. I'm going to show a video from the lab in a moment, but before we do that, I just wanted to have a very quick run through of the gene that we're trying to get into the cells here. So it's called tissue plasminogen activator, or TPA, and this is the experiment really. So we start off with the gene for TPA. We're putting it into a plasmid, which is a circular piece of bacterial DNA, otherwise known as an expression vector. And so getting that dark blue gene into the expression vector, and then trying to transform an E. coli cell with that vector. And if we've successfully transformed that cell, then it will then start pumping out the TPA protein, and we can then use that for a drug, because it's used as a drug for stroke patients. And here it is getting injected into the neck of a stroke, someone who's at risk of having strokes. So that's the idea of the experiment. And we'll now go over to the virtual lab, have a look at how this would work. And then afterwards, it's about a 6-minute video. Then we'll have a look at some results and interpret those results. Okay, so let's go to the lab. Whenever we genetically engineer bacteria to contain a gene of interest, there are really 2 steps in the process. First of all, we've got to try and get the gene into the cells. But because that isn't always successful, the second step is finding out which cells have successfully incorporated the gene. In other words, have been transformed. And what we're doing in this experiment is we're using E. coli, which is a species of bacteria, which contain a plasmid, a circular section of DNA, and we call that the expression vector, and it's into that plasmid that we're going to try and get our gene, the TPA gene. Now, some of those cells will be successfully transformed, and some of them won't. So the challenge is to distinguish between those cells which contain the original expression vector and the expression vector which incorporates the TPA gene. And the way we can do that is thinking about restriction enzymes. Now, restriction enzymes are those specific enzymes which can cut the expression vector at a particular spot, and they recognize a particular sequence of DNA bases. And if we use the restriction enzyme on these samples of bacteria, if the cells haven't been transformed, then the restriction enzyme will only cut at one place. Whereas if the cells have been transformed, the restriction enzymes will cut at 2 places and excise the gene that's been inserted. So we're going to use a gel like this one here to load our experimental samples along with some controls, and we're going to find out after treatment with the restriction enzymes whether we get one DNA sequence, in which case we'll see one band on the gel, or whether we see 2 sequences, If we see one sequence, that's the empty expression vector, and we haven't transformed those samples, and we can discard those bacteria. But if we see two bands on the gel, that means we've got the gene of interest, the TPA gene, in those samples of bacteria, and we can go ahead and use those cells, grow them up, and purify the protein that they're making, and use that for our drug. I've put the agarose gel into the electrophoresis equipment and covered it with buffer, and now the next step is for me to load the DNA samples into the little wells, the little indentations at one end of that agarose gel. I'm going to use one of these adjustable micropipettes with a disposable tip, using a different tip for each sample, and we're going to take those DNA samples from the ice bucket, pop open the lid of the tube, and sort of just inject each of those samples one by one into these wells at the end of the gel. The wells are on the right-hand side of the gel, and that's where the negative electrode is in the electrophoresis equipment. Of course, DNA is a negatively charged molecule, so when we turn on the power, the DNA is going to migrate from the right to the left towards the positively charged electrode. As the DNA moves, it's going to have to move through the agarose gel, which acts like a sieve. So the smaller DNA molecules, the smaller strands of DNA, are going to move more quickly through the gel and progress further towards the positive electrode. Conversely, the larger stretches of DNA are going to stay behind, moving less quickly. After about 30 minutes, we should be able to see our DNA strands in the samples sorted by their molecular weight. Now that the electrophoresis step has finished, it's time to visualize the bands in the gel. And of course, DNA is not visible to the human eye. It's not a colored substance, but there is a UV fluorescent substance in the gel which fluoresces when attached to the DNA. So we should be able to see that UV fluorescence in this cabinet here. So we're going to load the gel into the cabinet, turn on the UV light, and see what we can see. All right. So let's go back to, not there, let's go back to here. Okay, brilliant. So hopefully you could see me in the lab and understood something of what's going on here. And hopefully you can still see the screen that I'm sharing. So we've got this restriction enzyme, which is going to be cutting the untransformed plasmid at one site. It will cut the transformed plasmid at two sites. And we've run the electrophoresis experiment, and here is our result. And you see we've got a number of lanes in our gel. The first lane, we've got a lot of different DNA sequences in something called a DNA ladder. So this is a commercial sample of different lengths of DNA, And we've got an explanation of what's going on here, which is provided by the company on the right-hand side. And you can see the heaviest strands of DNA with over 10,000 base pairs are at the top, and the lightest are at the bottom. So in other words, we're loading the DNA samples at the top of this UV image. And so the negative electrode was at this side, the positive electrode was down here, and the DNA strands are moving from top to bottom, and the heaviest strands move the shortest distance, so they're nearest the top. That's the 10,037 base pair strand there, and the lightest ones go the quickest, and so we can see the 200 base pair strand down here at the bottom. So that's our calibration lane, our reference lane. And then we've got the experimental samples in lanes 2, 3, 4, and 5. What I'm going to ask you now is just to try and interpret these data by typing in the chat. Which of these samples, and you can just put in the numbers, which of these samples have been successfully transformed? So put in the chat 2, 3, 4, or 5. Which ones have been correctly transformed? And let's see what you get. Three and 4, brilliant. So we've got several of you who've got that correct. Very good. And the reason being that simply that we can see 2 bands in that in those samples. This band here, which is the heavier DNA sequence because it's nearer the top of the gel, is the vector. They've all got the vector, you see, even 2 and 5, which weren't properly transformed. They contain the empty vector. They've all got the vector, the expression vector, but it's only 3 and 4 which have got that that inserted TPA gene. Very good. So for bonus marks, my second and final question, because we don't have very long, my second question is, roughly how big is the TPA gene? Take a look at that image, see if you can match, just roughly match up how far that TPA gene has gone with the DNA ladder, and let me know roughly how big the TPA gene is in units of base pairs. This depends a bit on how good your screen is and how well you can see my screen. But just give me an estimate. All right. We've got someone getting 5,000 base pairs. We've got someone going for 400. There's quite a range there. We've got someone going for 2,000. Someone's 600. Okay, so everyone's answers somewhere between 400 and 5,000 so far. Let's see if we can count the rungs on this ladder. So it's 1, 2, 3, 4, 5, 6, 7, 8. So it's somewhere between the 8th and the 9th rung on the ladder. 1, 2, 3, 4, 5, 6, 7, 8, and 9. All right. So I think I like the 2,000 answers best. I think it's somewhere between 2,000 base pairs and 1,500 base pairs, because it's in between those rungs of the ladder. So we're lining it up on that calibration lane. And so somewhere around 2,000, or maybe perhaps 1,800 base pairs. So with this kind of experiment, I'm going to stop sharing my screen now. With this kind of electrophoresis experiment, we can not only tell which bacterial samples have been transformed and which ones to grow up and purify the protein from, but we can also, if we wanted to, make sure that we've got the right gene by measuring how big it is, because electrophoresis is such a powerful technique, such a powerful chromatographic technique, that we can measure the size as well as the number of DNA sequences. So, well, thanks very much for your attention. Thank you for those who answered my questions so well in the chat. And I'll hand back over to you, Latsi. We're just about on time.
Fantastic. Thanks a lot, James. And as close as we could get to the practical online, but I think some of this can be replicated in the school, and we are very happy to share the setup protocols and how to do, and also share some of the materials if some of the school wants to set this up. And so now we go to the second session of our talks and posters, and it still stays with molecular and biomedical topics. So just to show you the program here, and in this order we go to the aging, and then this is the heart transplant, which I already wrongly introduced with the previous poster. And then it will be an exciting talk about this microscopic level, the genetics, how far we can go with genetics, and then the posters. So let me go to the first set of talks, and the first is the aging. And so it is Jack, Roman, Keshav, and Chiba. So are you there?
Yes, hello.
Okay, hi.
So our presentation is on aging. Next slide, please. Senescence, or biological aging, is the accumulation of a wide range of molecular and cellular damage. This leads to a gradual and inevitable decrease in the faculties of an organism, via reproduction, respiration, or movement. However, these changes are not linear nor consistent, and are only loosely associated with the aging of an organism. Although there are many markers for aging, the most widely accepted marker is telomere length. Telomeres are protective caps on the ends of chromosomes, made up of thousands of base sequence repetitions, which is TTAGGG in vertebrates. Sheltering proteins bind to this specific sequence and prevent chromosomes from fusing together in non-homologous end joining and causing genomic instability. The natural DNA replication processes causes telomere attrition, and the telomeres eventually become so short that the cells undergo apoptosis or senescence, enforced by the p53 pathway. Next slide.
Telomere shortening causes mitochondrial and genomic dysfunction. For example, telomere shortening reduces the amount of binding sites for sheltering, which, via a complex mechanism, activates cyclin protein P21 and induces the production of reactive oxygen species. These highly oxidative species attack many DNA bases, causing genomic instability and hence inhibit DNA repair mechanisms. Exposure to RRS species increases the chance of mutation and hence increasing the chances of diseases like cancer. In addition, senescence by sheltering inhibition decreases the number of mitochondria produced, hence decreasing the efficiency of ATP production and causing metabolic dysfunctions such as neurological and cardiac diseases. Next slide, please.
So one promising route to stop or slow aging comes via epigenetics, which is expressing different genes. So in 2006, a Japanese scientist called Shinya Yamanaka discovered these 4 genes on the board, which managed to turn adult somatic cells back into embryonic stem cells. So these are called iPSCs because they induce pluripotency. And these iPSCs, they express telomerase, which is an enzyme that lengthens telomeres. Another thing that iPSCs do is they have significantly reduced mitochondrial ROS production, which, as we saw on the previous slide, is causes several symptoms of aging. Furthermore, in order to have more efficient iPSC production, the OCT4 gene is used to inhibit the p53 tumor suppressor pathway. As we'll see later, this has several side effects. So how are these Yamanaka factors used? They were proven in 2016 by Campo to be able to rejuvenate damaged tissues in live mice. And in 2020, they were shown to reset telomere length in human cells. Next slide, please.
The process of dedifferentiation closely resembles that of tumor formation. If unchecked, these embryonic stem cells can continually divide without shortening telomeres and without the p53 pathway to suppress tumors. Tumors can be malignant or benign and can spread to other parts of the body in metastasis, in particular the lymph nodes, which help fight infection. The suppression of action of these nodes can be severely detrimental to someone's health. However, if cancerous growth can be avoided, controlled use of the Yamanaka factors could exponentially increase the quality of life. Some people may argue against increasing lifespan and potentially prolonging suffering. However, researchers are instead trying to increase health span, which would mean that the aging population would remain healthy. On the other hand, it could be argued whether it's morally right to prevent aging, as issues such as overcrowding may affect generations. Next slide, please.
Thank you very much. Any questions? A quick question that so the telomere is measuring the number of cell divisions and sort of puts a limit to it. So is this a good news, a good mechanism, or is this something which, as you described, can be overcome and manipulated for the good?
Well, I mean, telomeres have evolved because they have a good benefit, obviously, which is to prevent cancer, to prevent excessive division. But of course, this comes with the side effect that as organisms age, they deteriorate. So if we do try to stop aging, it then puts us back into the risk of inducing cancer. So that there is a fine line to be crossed there.
Absolutely. Very nice. So it's a catch-25. Very nice. Any further questions? So if not, then thank you so much. And we will have a very, very related post as well in this session, poster 11, which I highly recommend to take and sort of read it together with this lecture. So then the next one is the development of heart transplants, and this is by Tyler, Mia, and Daya. Are you around?
Yeah, I'm here.
That's great.
So I'm going to be talking about the development of heart transplants. Can I have the next slide, please? So the origin of heart transplants. The first heart transplant was successfully carried out in 1967 by Christian Barnard in Cape Town, South Africa. Since that time, there's been little change in the surgical techniques that are actually used to carry out heart transplants, but there has been growth and change in immunosuppression, which targeted immunosuppression began in the 1980s. Next slide, please. So how is organ rejection prevented? So a big risk with any sort of transplant is that the immune system will reject it. So immunosuppressants are taken to weaken the body's immune system. This is important as the body will try to attack the transplanted heart because the CD4 plus T cells that detect foreign material will release antibodies that can damage the organ. So this is done through DNA methylation, which is a process where methyl groups are added to a DNA molecule. It's an epigenetic modification, which means that they affect gene expression. So steroids used to be used, but but they're not used as much anymore due to the high risk of metabolic, skeletal, and vascular disorders, which means drugs such as AZA and MMF have been developed as they have fewer long-term side effects than steroids. Next slide, please. So complications associated with heart transplants. So during the surgery, the patient is put on cardiopulmonary bypass so that the surgeon is able to take the diseased heart out. The longer that the patient is on bypass, the greater the risk of decreased heart function in the long term. When the surgeon reconnects the blood vessels to the healthy donor heart, they're under a very high pressure, meaning that the stitches may not hold. This means that the vessels may begin to leak, which can be life-threatening. The patient's immune system could reject the donor heart. To prevent this, the patient is given immunosuppressants, which help with rejection, but also make the patient more susceptible to other diseases such as anemia. After the surgery, there is a high risk of blood clots. These can travel to the brain, causing a stroke, or even to the lungs, which induce lung problems. Next slide, please. So the hope for the future of heart transplants is that 3D printing will be able to be used. So it's being researched as using artificial hearts would remove the need for organ donors and possibly increase the lifespan after surgery, which currently sits at around 13 years. So to create a 3D printed heart, ornamental tissue is taken from the patient. This forms a personalised hydrogel, which encapsulates cells, making a bioink formulation. This is printed to make the complex cellularized structures of the heart. The first successful heart made of human tissue was printed in 2019 at Tel Aviv University in Israel. Researchers estimate that a transplant using a 3D printed heart won't happen for approximately 10 to 15 years, as the 3D heart that has been printed is only the size of a rabbit's heart. It's unknown yet whether a printed heart the size of a human's heart would have enough strength and integrity and the essential pumping ability needed to work. So thank you.
Thank you so much. Any questions and activities? So I have one, because when I see such topics like printing a heart and reconstructing by engineering, I always think that this is a science fiction. So what do you think? Will we ever get to the point that we will have a transplantable printed heart?
I think we will. I don't know if it will be maybe even within our lifetime, but I certainly think it's possible because science and technology is advancing so quickly. So I definitely think it could happen eventually.
Absolutely. This is such a science at the edge. Yes, taking it to the total edge, but I am also firmly believe that this is going to happen. Maybe not with the heart, but other easier tissues first. Any other questions?
We do have a question in the chat, I think.
Okay.
Yeah.
So I read the chat from John Hale. Is there a different surgical stitch used on attaching the aorta and the pulmonary artery to cope with the pressure?
I've got to be honest, I haven't really looked into that. I think that Maya was maybe looking into that, but I'm guessing, I think the actual sort of suture technique is slightly different. I'm guessing they use a stronger material, but because the aorta is under such high pressure, I still think that it struggles to hold it together and cope with the pressure.
Okay, so James is leaving. Bye, James. So the next question is that would 3D printing be viable for replacing other organs and structures?
Yeah, so they've started researching into whether kidneys and even the liver can be 3D printed, and even ovaries. So they are printing ovaries that can restore fertility in women after things such as cancer treatment. And they've had high success in mice. They've already managed to restore fertility in mice, but they haven't moved on to human trials with that yet. So it's definitely like being researched.
Yes, and this partially also answers Miriam's question that that going to other tissues like liver is definitely liver must be a more solvable problem than the heart.
Yeah.
Thank you so much. So we go to the next one, which will be the culture on the microscopic level, and that talk is from Tiffin. So I maybe didn't mention that the heart talk was from Rugby High School, and this is Marta. So Marta, you are around?
Yes, good afternoon. We're here.
Yeah.
Can you hear me all right?
Yes, we can hear all of you. Thanks.
Okay, good afternoon. So we're going to be speaking about culture on the microscopic level and the influence of culture on genetics. Could we have the next slide, please? So an epigenetic modification is an addition to DNA, which will alter gene expression, but won't necessarily change the base sequence of the DNA. And through alterations in spatial arrangement, for example, or the addition of chemical groups like stickers basically to the DNA, the level of transcription and therefore protein synthesis can be modified for different genes. And there are two core mechanisms through which gene expression is altered: histone acetylation and DNA methylation, and they both work in different ways. And in eukaryotic organisms, DNA is tightly wound around large globular proteins called histones, and acetyl groups can be added by the very creatively named histone acetyltransferase enzyme. And the addition of acetyl groups will reduce the tightness of the association between DNA and histones, and this will make it more accessible to different transcriptional enzymes. And this will mean that there is obviously an increased rate of protein synthesis from that section of DNA. But on the other hand, we've got DNA methylation, which will suppress the expression of a certain gene by the attachment of a methyl group to the DNA-based cytosine. And it's attached to the fifth carbon, and this will form 5-methyl cytosine. This will attract proteins which inhibit the binding of transcriptional enzymes, and it acts as almost a blockade of the machinery that's required for transcription to occur, thus having the opposite effect to histone acetylation and reducing the rate of protein synthesis. Next slide, please.
So, lactose tolerance is the first example of how a culture shift has influenced the human genome. The rise of cattle and sheep as domesticated animals originated in what is now modern-day Turkey, Iran, and Iraq about 10,000 to 13,000 years ago. Over hundreds of years, human populations migrated west into Europe, and with them they brought their animals. Therefore, upon their arrival in Europe, they introduced the ideas of farming and domestication. Over time, due to the proximity of cattle, sheep, and farmers, the farmers eventually developed a mutation in their genes which allowed them to drink milk, as they could digest lactose. Through natural selection, the frequency of this mutation rose throughout the human population in Europe, and this resulted in the majority of people being lactose tolerant. Lactose tolerance is determined by the body's ability to digest lactose, and therefore the levels of the enzyme lactase. The production of lactase is a genetically determined trait due to cis-acting polymorphism. This is the process by which environmental and epigenetic factors disrupt or enhance binding sites during transcription, thus influencing the expression of individual genes. 13,910 bases from the transcriptional start site of lactase, a thymine base caused by substitution mutation, results in lactose tolerance, whilst the cytosine base here leads to intolerance. The presence of the thymine base results in increased binding of an activator, OCT1. This attracts more transcription factors to the lactase gene. Consequently, there is an increased rate of transcription for lactase, and steady levels of lactase are maintained in epithelial cells, specifically the cells lining the intestines. This allows for the successful digestion of milk. In fact, research conducted during the Genographic project showed that due to the high frequency of milk drinkers, most of the population of Europe today must have descended from cattle herders and farmers. Next slide, please.
Obesity arises from interactions between genetic, environmental, and behavioural factors. A study of male albino rats who had been fed with a high-fat diet and mated with female rats who had a normal diet showed that although the offspring had a normal weight, they showed abnormalities in the gene that controlled metabolisms. As a result, many of the offsprings and later generations showed symptoms similar to type 2 diabetes.
This shows that for humans, a culture of high-fat diets can affect the epigenetic markers on genes coding for metabolic hormones, as the methylation patterns in obese mice and humans are the same. Another study compared the sperm of 13 normal weight human subjects and 10 obese subjects. The sperm of the obese subject showed a distinct epigenome that characterizes human obesity, with distinguishing DNA methylation patterns, which would then be passed on to offspring and therefore implicates an epigenetic mechanism in sperm that would increase the likelihood of a child developing obesity later in life. This creates a positive feedback loop, as a high frequency of obesity increases the prevalence of epigenetic markers within a population. This in turn increases the proportion of obese persons, which has consequence on the average socioeconomic status due to social prejudices. A lower socioeconomic status is often associated with less healthy food habits. However, an understanding of the epigenetics of obesity leads to an increased knowledge of genes involved and can lead to more targeted treatments. One of the first genome-wide methylation studies revealed increased methylation at the UBASH3A gene and reduced methylation levels at the TRIM3 gene in obese individuals, providing evidence that obesity is associated with epigenetic changes. Thank you.
Thank you so much. So, waiting for questions, but until they arrive, just one note that one of my favorite scientists is Sidney Brenner, a Nobel Prize winner on the apoptosis and worked on C. elegans. So he is always writing provocative essays, and one of his essays he meditated that what are the diseases, and then he comes to that the disease is very much current days, that our genome is not in harmony with our environment. There is a mismatch between our genes and the environment. So I was puzzled by the title of your talk, but it now I fully get it, that something like this is the message, that we changed our environment so quickly. For instance, from hunter-gatherers, now we go to the supermarket to buy our food in the anything we can lift from the shelf, and many of our diseases coming from this, that we as humans changed our environment, but we are not adapted to it. Is that true?
I think it's definitely true. I mean, if you look at the Dutch hunger winter as well, that was a very quick kind of environmental change. And the genetics of kind of people living in the Netherlands today are still being studied, and we're still seeing the effects of the hunger winter within their epigenetic code. So definitely kind of quick changes in the environment can have really big kind of epigenetic changes. And I think we need to move away from thinking that it's just mutations to DNA that are the issue. It's also epigenetics as well.
I see. So these two factors hand in hand puts us in trouble to respond to our environmental change. So it was a very nice talk. Thank you so much. And I think this was our last talk in this session. So now we go over to our posters, and let me pick them up again. And so the first was the cholera, and so this is a poster which really works because attracting the attention, but it's also very informative. So I think it's a beautiful example for a poster, and it is about cholera, and nicely describes what cholera is. It's a bacterial pathogen, and the problem with it is the toxin which it secretes, and then how it acts is acting on a signaling protein and the signaling compound production called CRMP, and that will put us in trouble because of the homeostasis with the sodium and the chloride channels, and so the salt homeostasis. and that will lead to the sudden loss of water from our system. This part shows that this is a huge problem which could be solvable, because this is a pathogen which we could definitely combat. But the poorer parts of the globe is really exposed in a unbelievably and unfairly high level. And there are simple solutions such as clean water, believe or not, washing our food. But these people unfortunately don't have access to these basic commodities. And the treatments as well. We know that we don't have this disease, or very rarely, in our part of the world. So clean water and sanitation and nutrition. So this is a very nice poster. Thank you so much. Any comments to this? Please put it also on the Padlet comment box that we use it. So then our current problem, which again is going back to infectious diseases and our current problem with the pandemic and the COVID situation. So this is a poster which extremely nicely explains us how vaccines work and will produce the immunity. And specifically, this is an example of the RNA vaccines. So believe it or not, this is a new invention which pushed its way to this fast application just because of the pandemic. We didn't have RNA vaccines before, but it happened to be one of the best routes to produce vaccines. And the RNA is injected to our muscle, which gets to our cells and gets translated. And the spike protein, which the RNA codes for, will raise the immune system through the B cell immunity and production of antibody and producing these memory B cells as well. And so that also goes back to the topic of this herd immunity, that the more of us are vaccinated, either naturally or through the vaccines, the less the pathogen has ability to spread. So this is a nice diagram showing how that works. And also this patience we need now that we cannot leave the lockdown. Sorry, I got muted, but took time to navigate me back to that thing. So it would be good if please don't play with that muting each other. So let me go back to the poster. So we were with the vaccination. So the next poster is the tobramycin, and that one is number 10. So this one is talking about a disease which is cystic fibrosis, and this is a genetic disease where we know the genes which am I muted again? No.
No, you're not. We can hear you.
Please let me know if you can't hear me. That then I or try to unmute me. Maybe you can also do it. And so this is a nice post describing the disease, and it's essentially again an engineering trick that what the disease causes, that it's making development of a mucus within the lung surface of the alveoli surface, and then a bacterial infection which develops this unpenetrable barrier, and that makes the treatment very difficult. So here the trick is that they developed a system where they coated the antibiotic in a sort of a nanostructure which enables to penetrate this mucus and make this antibiotic work better. So it's an engineering hand in hand with biology. So very often multidisciplinary is another key, that studying physics together with biology makes a lot of sense. So this is the poster, very much goes back to the telomere problem which we heard from the talk, and very similarly and nicely describes that every cell division the telomere shortens, and shortening telomere will cause the cell death and essentially preventing the further proliferation of the cells. And again, it is about an enzyme called telomerase, which adds this end bit of the chromosomes, which explained nicely here. And then the next one actually came as a talk format, but it is about the COVID mutations. And as we know, mutations generate the variant of this virus, which puts us in a lot of trouble in terms of the effectiveness of the vaccines if that would be overcome. And this is an RNA virus, so that's why the mutations are even a higher rate, and makes with RNA viruses is always more problematic that the mutations are higher in a higher rate. And this is the nature of the virus, so we cannot do anything about it. And some mutations are without effect, while other mutations will make the virus more virulent or more resistant to the vaccines, for instance, or even drugs. And so then this is another interesting post, and I forgot to tell the schools, but this is from St. Bernard, from Jessica and Ria. And it is about the problem of the newborn babies and connected to the development of the lung, that this alveoli in the lung in a very early development, they are not able to keep the shape and collapse, and it goes to a treatment that with surfactants they can make a little coating within the lung, as I understand it, and that will protect this collapse from the pressure in newborn babies. So essentially, this sort of simple idea now saves a lot of newborn children, if I am right. Any comments? Because I am very far from biomedical science. I am a plant scientist. So I might be telling rubbish here and there. So please correct me. And the last in this session is about peanut allergy. And we know that this is a big problem for some people and the allergy reaction can be so severe that it can be life-threatening. Therefore, the labeling of the food needs to be very strict. And I am sure you picked up cases in news where accidents happen. And this is about a, a protein within the peanut which the immune system in some persons reacting to, and overreacting and bringing in this hypersensitive reaction of the immune system, allergy. And so this poster is about that if we train the immune system by low doses in a continuous manner, and in this example putting it in a toothpaste, then this educating and training the immune system and slowly developing, not in high dose but in slow dose, our memory B cells, which can react, and we don't have this overreaction and the allergy response that can help. So this is a simple idea. Sometimes simple ideas can go very, very far, and this poster is describing that. So I don't see any comments. So then I think we finished early, unbelievably, with this session. So now the question is whether Enrique is already in our call. For our second practical. If not, because we have 10 minutes, so if Enrique is not yet in the call, then we continue, I believe. That's what I would suggest. That Enrique, are you in the call? Maybe not yet. That. I suggest then that we go with some further talks, and then when Enrique joins us, we will have the second practical. So we continue with the session 3, and session 3 we have the talks which now more oriented towards the ecological problems, but the first talk is still cancer related, but it is framed in a bit of an ecological wording, which asks that why large animals are less vulnerable to cancer, and then we go to the bees. And so if Jack is around.
Yeah, hello, can you
Hello,
hear me?
Jack. So then I bring up your presentation. Okay, so here we are. So Jack is from the Richmond School.
Hello, everybody. Today, my talk is about cancer and cancer's tendency to not be as prominent in larger animals. Can you click, please? Can you click, please? Thank you. So my talk was something which I find really interesting, and it's the whole topic of Peto's paradox. Now, if you're unfamiliar with what it is, I'm going to go over the whole premise of Peto's paradox and why it poses such a huge issue when deciding the cancers. So would you mind clicking? It is a bit reductive, but it can be said that since cancer is just an error in mitosis, we can reduce the rate of cancer to a mathematical probability. But why is this important? Would you mind clicking again? Thank you. Smaller animals do have less cells than larger animals. It can be said that animal cells are of similar size. That's not, of course, to say that nerve cells are the same size as liver cells, because that would be unfair to say. What I mean to say is that if a crocodile's liver cell is of roughly the same size as a monkey's liver cell, there is just more of them in the crocodile. Would you mind clicking again? Now, why is that important? Well, the more cells you have, the more mitosis that you have to do. And therefore, linking back to the statistical probability, there is more chance for cancerous growths to form. Well, that is in theory. Would you mind clicking again? This does not happen in actuality. Larger animals should have a higher proportion of cancerous growths, but they don't. It's the smaller animals who are more susceptible to cancers. So why is this? Well, in this talk, I'm going to go over 2 of the leading probabilities for why this happens. Would you mind clicking? Thank you. Explanation 1 is cancer's innate abilities and its own selfishness. In an overarching, it is the cancer that is killing cancer. The second argument is in evolution and how it is more evolutionary significant for larger animals to have cancer suppressants. Would you mind clicking? Thank you. Now, I'm now going to be talking about cancer's self-destructive properties. Would you mind clicking again? Thank you. Maybe a bit simplistic, but smaller animals are of less mass than larger animals. Would you mind clicking again? This means that if you were to get a 2-gram tumor in a monkey, that 2-gram tumor is 10% of the body weight of the monkey, whereas the same 2-gram tumor is only 0.002% of the elephant. Would you mind clicking? But why is that important? Well, would you mind clicking? Thank you. Cancer cells are unstable, and their selfishness makes them expand uncontrollably. Would you mind clicking again? This expansion is only stopped by the cancer's ability to gain energy for growth. By that, I mean its ability to get oxygen and glucose, things that you need to undergo mitosis. Would you mind clicking again? So, because cancers want to grow constantly, they do something really clever called angiogenesis, which basically is just the splitting of blood vessels. And what it means is that in theory, cancer should be able to grow unchallenged because it has an unlimited supply of food. Would you mind clicking again? Now, because of this, in theory, endless nature, the cancer cells will continue to grow. However, cancer cells are selfish, and each replication has a chance to sever communication and become its own tumor. Would you mind clicking again? Now, this separate growth is going to compete for the food and the resources that are available with the parent tumor. Would you mind clicking again? This will also call for angiogenesis, which is again another splitting of the blood vessel, until there is not enough space for both tumors and they both eventually die. They might not die in completion, but it will drastically reduce the amount of tumor there is, which again is very useful. Would you mind clicking again? This second tumor is called a hypertumor, and there are ways to increase the likelihood of one. For instance, phosphorus. The more phosphorus there is in the body, the more chance of a hypertumor of forming. But if you think about it on a simplistic level, larger animals have more mass. That means that a tumor needs to grow larger before it has an effect, which means there are more chances for hypertumors to form. Now, there are obvious problems with this hypothesis because the size of the tumor does not inherently mean its toxicity. You can have the same tumor, have the same lethality, but it's much smaller. But it is an explanation, and it has been one for quite a while. A newer one is the evolutionary argument. Would you mind clicking? Now, I quite like this one. Would you mind clicking again? Thank you. If you think about it basically, the main aim for evolution is to create offspring. The main aim of an animal is to produce offspring, and this is known as fecundity. Would you mind clicking again? Larger animals, on average, require more time before they reach sexual maturity compared to smaller animals. Would you mind clicking again? Thank you. This means if you think there is more evolutionary incentive for larger animals to have more cancer suppressant genes because they have more time before they reach sexual maturity, so cancer is more of an issue for that as that decreases the fecundity. This is compared to smaller animals who there's less biological incentive for them to have cancer suppressant genes. They obviously still have them present, but they're not in as great number. Would you mind clicking again? Thank you. Due to this, there are larger species tend to have more cancer suppressant genes to ensure correct replication of oncogenes, which are just basically genes that have the potential to cause cancer. And an example of this is a TP53 gene that is found in elephants. Would you mind clicking? Now, this is a real experiment which I thought was really cool, and that there is a type of virus called an endogenous retrovirus, which once integrated into a DNA can cause cancer. So a study was done with 33 mammals, and it found that mice, which was used as a small animal for this study, there was 3,341 samples of the retrovirus DNA in the genome of the mice, whereas dolphins, there was only 55 virus samples. This basically means that because there are less virus samples present in the dolphins, it means that there must be more genes there dedicated to stopping these cancerous growths, because once a endogenous retrovirus is present in the DNA, it causes cancers. So that means larger animals have this mechanism because there's less of the DNA present in the genome. Would you mind clicking again? So why is this important? Why does that matter for the future? Well, would you mind clicking? Well, for a start, paradoxes, I think, as a concept, prove that science is still exciting. We have made great leaps in scientific advancements, but paradoxes and issues like this still prove that there's some aspects of the natural world we don't understand, and there's still a plethora of discoveries that we can still make. Would you mind clicking? Another important thing is that despite the fact there is no definitive answer, I don't think something needs a definitive answer to be useful. I think that this starts a conversation between intellectuals, which can only reduce into a deeper and more fulfilling understanding of the topic. And once there's debate amongst scientists, that can only lead to a positive outcome. Would you mind clicking again? Yeah, by understanding more about how cancers form in animals, not only can we prevent the growth in larger animals, we can apply the information to humans, setting up a field of cancer research that could help us understand about how a species could be implemented preventations for cancer. And the last one is that this allows us to challenge causation of cancer, not only on a large scale, but on a cellular level. So we can see how cancer operates on an individual scale and allows us to create the concepts such as hypertumors, and makes us understand more about how cancers form, how they come to prominence, and how maybe we would be able to stop it. Can you press, please? Thank you, everyone, for listening.
Fantastic. Any questions, please? I have one, just until we get from others that so you presented very different solutions to this paradigm. So the question is that so the last paradigm which you seem to be the last solution you seem to be the most preferring is this tumor suppressor genes are more in the large animals than in the small ones. So can we answer this by looking at the sequence? So we know the sequence of the elephant, the genome, the entire genome, and the sequence of the mice, the genome sequence. So do they have more of these tumor suppressor genes?
From the research I've done, the larger animals do tend to actually have more tumor suppressant genes than smaller animals. An elephant gene would have more tumor suppressant genes than that of the mouse. It was more why that I found very interesting.
But very often then the argument would be that if you are looking for something in biology, you will ought to find it. So how could we prove it definitely that that this theory is right?
Well, how we could prove it definitely is by looking at known sources of cancer. So like the study which I gave, an endogenous retrovirus is a known cause of cancer, and that by having them integrated into the DNA, into the genome of the animal, it proves that it has had that mix with this virus. And the more times that there are present in the genome, the more times this virus has been through evolution, been in this animal. So the fewer times that it's present in the genome, it can be said that there are fewer times the cancerous causing thing has been present in the animal, meaning at some point there would have been an adaptation to make that cancerous causing thing less able to get into the animal and be able to inject itself into the DNA. So the less numbers there are, the more suppressant, the more mechanisms they have to prevent against these cancerous causing things.
Yes, so it's very clever that so you don't do an experiment, but you are following the footprint of the cancer causing phenomena, the virus in these animals, and that footprint tells you the causal effect more or less. So that's a very clever idea. Any further question? So we have one from Enrique. I can read it out. At least we know he's online now. That select the tumor suppressor gene family, identify all the homologs in the genome of 10 chosen species, and plot the numbers found against the size of the animal. Is there a correlation? So this is Enrique's solution. Yes.
Yeah, that's a good solution.
Okay, so I think now we go back to our program and have a bit of breakaway from the talks with another practical. So I unshare my screen and give the screen to Enrique.
Yeah, hello. Thanks very much, Lassie. Can you hear me?
Yes, we can.
Yeah. I'm going to try to. Yeah. Can you see my screen?
Yes, we can.
Thank you. Yeah. All right. So let me see. Let me see how this works. Yeah. Yeah. Good evening, everybody, and apologies. I am actually in an office not far from our lab, and been a few times back and forth. So the idea of this practical, this is actually part of a practical, or the core of a practical in a second year plant life course in our biology degree. Other people also take it, biochemists, ecologists can choose it. So the question is, can plants see? Why do they see? And how do they choose how to flower? So let me tell you a little bit what the problem is. Let me show you how a solution to the question might come from using genetics, what we do in the lab, what sorts of data can be collected, and how that is combined with what we call using genetics, identifying the mutation in individual mutants, and try to see whether the two things combined give us an answer to the problem. So let me tell you what the problem. When people come to the lab, they are met with plants which are flowering. They are little plants, little weeds. They are common, not terribly common, but you may have them in, for example, in a patio, in your garden at home, on verges by the pavement, etc. It's a little thing called Arabidopsis thaliana, and it was the first plant for which the full genome was sequenced. If you want, it is to plant science a little bit what Drosophila, the fruit fly, is to genetics, or mouse is to biomedicine. So this is the plant. It produces a few leaves, very, very small, close to the ground, and then at some point it flowers. And when it flowers, it is producing those little flowers. It produces a stem to put them up high. Now, that plant has flowered at a particular time. These other plants seem to have either not flowered or flowered possibly at an earlier time. You know, they seem to be doing more than this plant has done. Are these changes in differences in when these plants have chosen when to flower? And what is involved in plants choosing to flower? Let me see whether this video plays. So at the tip of a plant, in between, at the tip of a shoot, there's a little pool of what we call stem cells. In plants, we call that pool of stem cells a meristem. And they produce new organs continuously. So this has produced 2 leaves, then 3rd, 4th, here comes 5th, 6th. There's a tiny pool of stem cells dividing there and producing new leaves. And at some point, they stop producing new leaves and begin to produce something, this thing which does not quite look like a leaf anymore, but this does actually, this is now opening and seems to be basically a flower bud. You see, those are flower buds now. So what's happened here is that that pool of stem cells, if you're on that growth tip, has changed what it was making. So basically, this is now a microscopy view of that shoot tip. And these are the leaves it was producing. And somehow, at the time when it was just producing leaves growing, it was at a stage that we call vegetative. Somehow it switches commitment or destination of the organs it produces at the sides. And now what it produces, these are buds of flowers. Yeah, from leaves to flowers. Now, how does this decision of what organ those cells, those stem cells are producing, whether a leaf or a flower, how is that decision taken? It's taken by sensing environmental conditions. For example, flowers mostly happen in spring, don't they? Somehow plants have to sense, oh, it is time to flower.
those pools of stem cells are taking decisions, and those decisions have to be driven by measuring, looking at the environment around them. How do organisms respond to their environment? We hear often this tension between, is it the environment, is it genes? Well, how do we sense our environment? Let me play you a little video of this amoeba. Is a kind of amoeba which is called a social amoeba. Dictyostelium, my colleague in the department uses is their organism in their lab. It's called Dictyostelium discoideum, and it moves towards a source of a molecule called cyclic AMP. Did you see that? So there's a little pipette here, full with a concentration of cyclic AMP. The video is played very fast, but the cell moves towards it. You can drive it mad if it could think. So how is the cell responding to its environment? Well, clearly it has a nose for cyclic AMP. So the responses to the environment are mediated by, let's call it a nose, let's call it a chemotaxis receptor, the products of genes. Ultimately, it's about genes. And plants see their environment because they have eyes, which are made, they're the products of the action of genes. We know that plants have eyes because we've all done this sort of thing, put some seeds to germinate in a dark place, some cress seeds, and we have these bean sprouts. The same thing on a windowsill produces normal seedlings. Plants have been aware of whether there was an eye. They must have eyes. And indeed, they have eyes, which we call photoreceptors. They are proteins. They're the products of genes. And there are two groups of photoreceptor genes that are particularly important, particularly well studied, and which we'll use in this practical. One is called phytochromes. We will look at one phytochrome. It's a protein which senses light primarily in the far red end of the spectrum. And the other is a cryptochrome. Again, there's more than one, but we'll look at one cryptochrome, which senses light, is another protein, and it senses light in the blue or blue UVA end of the spectrum. So when people come to the lab, if you had come to the lab, you would have found some plants. These are plants like the first we showed you. These are the population of plants that the three at the beginning were taken from. So these are normal plants. These are wild types. 28,000 genes working in these plants. The genome of this of Arabidopsis carries around 28,000 genes, similar to us. This plant, one of these plants, or this population of plants, all siblings, carry a mutation in one gene for one of these photoreceptors. They are mutant. So they have 27,999 working genes and one gene A faulty. So they are semi-blind. They carry a defect in one of these eyes, one of these photoreceptors, and they're semi-blind. And as a result, they're flowering in a different way. And these other plants are also mutant in a different gene. They also have 27,999 working genes, including the A gene, but have a different one faulty. They are semi-blind in a different way. And they are also flowering differently. So what you would have done is you would have come to see these plants and quantified how they are flowering. And what we do for this is we count how many flowers they had, how many leaves they had at the time they flowered. Sorry, forgive me. Oh, sorry. I probably need to reload this. Forgive me. So here we go. So you would have come to the lab and then, sorry, G. This is part of what our students had to see first before they came to the lab in our term 3. Yeah. So these plants, there they are, growing, flowering, da. So forgive me 1 second. Can you could you tell me whether you can hear the sound?
No, Enrique.
No. Forgive me. I'm going to stop sharing for a moment and reshare. And I think then, forgive me. No.
I did upload it on Padlet, so if we don't manage to show the video, they can watch after this film.
Okay. So we look at the flower. We look at the leaves. We count the leaves in the rosette at flowering. Yeah. I think there is sound now, but let's carry on. So then people count the number of leaves when the plant has flowered at different in the 3 genotypes in a sample of leaves each, and then they put the data on a document, on a Padlet document, and sorry, not on a spreadsheet. And let me see. Sorry. So it's on the way. So people put the data here, yeah? People put the data here. And what do we do with that data? Well, let's calculate some averages, some whatever. Yeah. And this is what we see. So the wild type flowered with about 8 leaves at the bottom. Mutant A flowered with far fewer, and mutant B with more. We can do a little bit of statistics, you know, these comparisons of means, etc. These t-tests. Oh yeah, those differences are significant. So those differences, what are they due to? Well, we would tell you, we tell people, one of those mutants, one of those groups of plants, the plants are faulty in one phytochrome gene. They have a change in one of 5 phytochrome genes, and we can actually tell whether a plant carries or doesn't carry that mutation, and this we will call genotyping. So we would take a bit of tissue of the wild type, a bit of tissue of each of the mutants, and extract DNA. Then do PCR, amplifying a bit of the DNA that carries, that codes for this phytochrome gene. When we do PCR, you know about PCR, polymerase chain reaction. Basically, we're generating many copies of that gene, and of exactly that region, nothing else. And once we have many copies, we can use a little trick. The sequence change in the mutant results in a pair of molecular scissors that we call a restriction enzyme, which can cut the wild type, becoming unable to cut. So if we take the PCR products and we subject them to the scissors, to restriction digestion, the wild type will cut, the mutant will not. How can we see whether it has or it hasn't cut? You have seen earlier a little bit of gel electrophoresis. We run them on a gel, a larger fragment, which has been not cut, will not move as fast as a smaller fragment, which will move faster through the matrix. And forgive me, I can't see my icons. Here is a little bit of what that isolation, if you want, was about. So
yes, now we are going to
so leaves are collected, then
we'll put the sample on ice for 5 minutes.
DNA is extracted by crushing in the presence of liquid nitrogen.
Now quickly vortex.
Then let's say PCR is done. Now we extract the DNA. We put some primers, some nucleotides, polymerase, etc. Put it in a machine that ramps the temperature up and down, up and down, up and down.
So we are going to take.
And then we do the digestion, and then we load the products on a gel. Yeah. And eventually we put
so these are the most
the gel on a gel dock. Look at the picture, and there it is.
You can zoom in basically.
There it is. So here it is. We do this, and this is the result. Now, can I please ask you? I will ask Professor Boger if he doesn't mind, so that I remain on the presentation, if he could tell me whether there are answers. So let me go back to slides. So the wild type product, PCR product, will be cut and will run far. The mutant PCR product will not be cut and will not run as far. We do two assays, one for a cryptochrome gene, the cryptochrome 2 gene, Cry2, and one for a phytochrome gene, the 5B gene, the phytochrome B gene, 5B assay. Could I please ask you which of these plants, A, B, and or wild type, well, the wild type is wild type, which of the two mutants, A or B, is the Cry2 mutant based on this result? Could you please tell us?
So I wait a little bit until more people vote. But we got our first vote is that the Cry2 is the B and the 5B is the A. That was the first.
Yes.
And let me see.
Do you agree? Do you disagree?
Is the B.
If you agree, you can put a thumbs up to the first answer or something.
Enrique. So
yeah, okay.
Do we
Sorry,
agree?
please. Yeah, we agree. So mutant B was the Cry2 mutant, and mutant A was the 5B mutant. So B is a cryptochrome mutant, A is a phytochrome mutant. Now, the funny thing is they are both faulty in a photoreceptor, they are both semi-blind, and yet one flowers quicker, one flowers slower. So let's see where we were. We said A and B. A flowers quicker with fewer leaves, has been flowering for longer, and that's why it's taller. It has more flowers. These are just starting. B flowers slower. When we took the picture, it had not begun. So it has produced more leaves. It's flowering more slowly. And we have said that, this plant was 5B, and this plant was Cry2. Now, what does this mean about how plants use light to decide when to flower? Let's think a little bit. How do plants tell when to flower? They tell the season. I won't play this, but we know this, yeah? We are somewhere up here, yeah? This would be winter. when the sun is coming this way, Britain spends some time in the light each day and a long time in the dark. Some time in the light, a long time in the dark every day. Days are short. This is winter. This is December. At the same time, in the southern hemisphere, in December, each day has a lot in southern Australia, let's say, or in New Zealand. Each day has a lot of light and a little bit of dark, and a lot of light and a little bit of dark. The days are long. They would be experiencing their summer, the December summer, and the days will be very long. So the day length changes with the seasons. Now, how can you tell the season? How can you tell seasons? Well, you need two things. You need a way of telling light and a way of telling time, which is independent of light. So you get up at 5 in the morning, you see light, oh, we must be in summer. Regardless of whether you have a calendar, you know it's summer. As long as the watch tells you 5 a.m. and your eyes tell you light. You need, of course, to have a watch that doesn't change the time in winter or summer, that says 5 a.m. regardless of whether it is summer or winter. A light independent endogenous clock. Now, do plants have that? Absolutely, absolutely. Look in some plants' leaf movements. Look what they do if they have been in light, dark, and then you put them in continuous darkness and you film them with a little bit of infrared light. The leaves go up and down and up and down. Sorry, let me go down. So they are waking up and going to sleep. Regardless of whether there is light, they do have an endogenous clock. And we have just seen they do have we know they have photoreceptors. So when you have the two, you can tell the season. How would you tell it's summer and time to flower? The clock tells you 5 a.m. or 9 p.m. and the photoreceptor tells you light. And you conclude, summer. So if you flowered in summer and your photoreceptor doesn't tell you light, what will happen? You will not be able to tell the days are long, so you will not flower. Therefore, a photoreceptor that is used to sense long days, when it reports to the clock there is light and is faulty, will result in a plant that does not transition to flower, that stays vegetative. What can we tell from our experiments? Probably this photoreceptor is a cryptochrome. Why? Because our cryptochrome mutant has not been able to transition to flower on time. It was behind, it was in the light, and it wasn't aware that it was in the light. Why would then a plant, which is semi-blind, flower early, as the phytochrome mutant flowered early? Actually, plants also use photoreceptors to tell other things. For example, to tell them whether they have plenty of light or whether they are in a shaded environment. They can tell whether there is enough light. They can even tell the color of the light. The light under a tree changes color, and plants can see this. It's more far red light rich, and plants can see this. And they see this because far red light inactivates phytochromes. And if the plant has no phytochrome B, it tells, oh, I must be on the very deep shade. I must run away. And one way in which they run away is they flower. They cannot grow themselves, so they hope for the best for their children. They flower, they produce babies, they produce embryos, pack them in a seed, and send the seed away.
Enrique.
There we go.
Thank you so much.
So,
we are very much overrun. We need to make sure we finish before the football starts. So I think time to push on. We had one question from Jon. So can you answer that, Enrique, in the chat? And we keep going if people don't mind. And
great.
So now we come back to our talks, and now this is the talk which I was alerted, but then we found it in the program. So we go to the bees and pollination, and that will nicely link with the talk in the masterclass from Ellie. So let me bring that up, and hopefully we still have Renuka. Renuka, are you there? Oh, was she already?
Yeah, I am.
Ah, you are there. That's great. So we can start then. Hello?
Just give me a second, Professor.
Okay, all right.
Evening, everyone. Today we'll be talking about buzz pollination and the decline of the pollinator. And yeah. So bees produce vibrations in a variety of behavioral contexts, including communication, defense, and pollen collection. And in the diagram, you can see that we've got the mechanism and the body parts of the bee and how they vibrate to get their pollen. So the pollen is collected using thoracic vibrations of the bee, and they shiver their flight muscles, which loosens off the pollen, and they collect the pollen onto their hind legs, so they're quite strong.
So the reasons why bees are declining are because of habitat loss, such as less forage and trees and shelter for the bees. Climate change disrupts bee behavior, and pesticides harm bee health. In addition, pests and diseases diseases that affect honeybees are shown to spread to wild bees, and managing honeybee health is one way beekeepers can protect wild bees as well as their hives.
So are we going further?
The other person in our group has some technical difficulties, so we'll just carry on.
Okay.
From here.
So the next slide.
Yeah. So in terms of habitat loss, how we can prevent the decline of these bees from natural predators is there's an organization such as Friends of the Earth, and they're calling for a sustainable approach to farming, such as integrated pest management, and one that will rely on less chemical pesticides, which are the primary cause for the decline of bees. And local authorities also have an important role to play as they manage significant areas of land, including parks, allotments, roundabouts, and road verges.
Researchers such as Jez Ross, he was passionate about halting the decline of British native bees, and he was working with big brands to encourage them to sponsor a hive, learning about beekeeping and selling their honey. And is crowdfunding to sponsor 250 hives over the next 5 years to breed British native bees.
So in terms of how tech is helping, there are a lot of new inventions coming out, such as microphones and advanced sensors that capture data from the hives and see invasive predators. There's also the use of AI that process data that has been collected to look for any anomalies to bee colony health. And in the future, we'll be able to prevent losses or mitigate problems that arise, hopefully.
Next. Are you ready?
Yeah.
Okay.
Robotics can also play a role, such as the digital bee being able to direct other bees towards nectar and pollen, and also help them navigate away from dangerous areas that contain pesticides or other obstacles. That is still ongoing at the moment, and hopefully in the future you should see more of those robotic bees. Also, in an effort to eradicate predators and mite infestations in bee colonies, scientists and beekeepers are turning to solar power. So the thermosolar hive uses solar power to heat up to 40 degrees Celsius for periods of time. And the temperature isn't harmful to bees, but is able to remove the mites. And this solar power technology is environmentally friendly, it's not harmful to the bees, and it's preferable to pesticides or other chemicals to which mites are becoming resistant. Thank you for listening. I hope you enjoyed our presentation.
Thank you so much. Beautiful drawings again to lift our spirit up. Thank you so much also for the content. So any questions?
Thank you.
Of the bees? Yes, our spirit needs uplifting, that's for sure. Yes, and it is listening to these nice talks, that's the way we get motivated. And so just one question, that this again is catch-22, that we need more food, and more food requires that we protect our plants from eaten by insects. And one of the main ways is spraying with chemicals, unfortunately. So can this be solved, that we don't kill the bees, but able to produce more food?
In terms of pesticides, there has been a movement to remove harmful pesticides like neonicotinoids that affect the bees' behavioral side of things in their brain, which makes them like dizzy and not able to fly straight and stuff like that. So there has been a movement towards pesticides that are less harmful and possibly not using pesticides at all in the future.
So one solution which scientists did come up with is that put the pesticide inside the plant by genetic modification. So this pesticide is a natural pesticide produced by a bacteria called Bacillus thuringiensis, and it's a peptide which kills the insects. So it can be put in the leaf, for instance, and only be expressed when the insects are biting. So, but unfortunately, a lot of people are not friendly with the genetic modification. So what you think? Would it be better to protect our plants through this GM route, or should we spray them with insecticide?
Definitely, genetic modification will help. And because of the new technology coming out, genetic modification will hopefully become easier and help, as it's already being used in food, such as I believe there's a yellow golden rice that's been genetically modified. So in that way, I feel that pesticides can also be genetically modified into the plant and cause less harm.
Yeah, I fully believe as well. And this route is already used as well all over the world except Europe. So now it might be possible after Brexit that it will be used in the UK. But we need to go with rationing, like this nice poster, that we need food and there is no good or bad. We need a solution to the problem solvers. That's great. Thank you so much. I don't see any further questions, so we go keep with this big problem, the bees and poisoning our bees, and therefore our food is compromised. So we go to the very same topic with a different angle. So the effect of cyanogenic glucosides on honeybees, and this is Loreto College with Emily and Daria. Are you there?
Hi, yeah.
Hi. So we can go ahead. So let me tell when you are ready.
Yeah, we're ready.
So start
Do
then.
you have the presentation? The PowerPoint isn't showing up.
So did you see the other PowerPoint then? Was it not shared?
Yeah.
Or it just stopped sharing? Let me go back and share. I don't know how it could stop sharing. Was I sharing the previous presentation?
Yeah.
Okay. So then, okay.
Okay. Hi, I'm Millie, and I've been working with Daria on this project. We're from Loreto College, and our focus for this presentation will be the effect of cyanogenic glucosides on honeybees.
Could we have the next slide, please?
I am on the next slide.
Thank you. Thank you. Cyanogenic glucosides are essential as a defense mechanism against herbivore consumption in plants, releasing toxic hydrogen cyanide when consumed. Hydrogen cyanide is an inhibitor to cellular respiration, preventing aerobic organisms from obtaining the necessary energy for function. The main symptoms for cyanide poisoning are difficulty breathing, fast heart rate, which if not treated quickly can lead to cardiac arrest and death. Plants also produce alkaloids, which produce a bitter taste, which acts as a natural pesticide against insects. This deters future insects from consuming the plant. The plant itself is not harmed, as the cyanogenic glucosides are kept in a vacuole, separate from the cyanogenic glucoside hydrolyzing enzyme called B16-glucosidases and hydroxynitrilases.
Plants must attract pollinators as they are unable to self-fertilize. Of the world's 115 most important crops, 87 require pollination to produce fruits, nuts, and seeds. Honeybees act as pollinators in biotic pollination, where pollen grains stick to the hairs on the legs of honeybees and are transported from the anther of one flower to the pistil of another to complete fertilization. Bees use protein-rich pollen for their physiological development as it contains essential amino acids, lipids, and vitamins. Nectar and pollen may also contain secondary compounds such as cyanogenic glucosides.
Could we have the next slide, please?
I am on it.
Thank you. Despite the toxicity of these secondary metabolites, honeybees aren't deterred from collecting pollen or nectar from plants. In fact, increasing evidence suggests that these toxic compounds may protect the bees from disease or predators, therefore deepening the already vital mutualistic relationship. Two methods of detoxifying hydrogen cyanide have been shown in living organisms. Rhodonase activity is found mainly in vertebrates, while bee synthase is found in plants and many insects.
Rhodinase is a multifunctional enzyme but plays a significant role in cyanide detoxification. One rhodinase gene was found in each bee species. However, B-cyanidine synthase was not present in the bee genomes. Bees have beta-glucosidase, which hydrolyzes glycosidic bonds, so it can break down cyanogenic glucosides into hydrogen cyanide. Accordingly, ingested cyanogenic glucosides do not appear affected by the beta-glucosidase activity found in whole bees and bee guts. The beta-glucosidase enzymes are probably either compartmentalized away from ingested cyanogenic glucosides in the gut or are ineffective during digestion.
Finally, we would like to say thank you for this opportunity to present our findings, and thank you.
Yes, thank you so much. It was a very beautifully put together presentation. So, any questions, please? So, I don't see any yet, but just one note that so plants are fascinating also because of the myriad of compounds they are able to produce, and in that way direct other organisms like bees to do some jobs for them or protect themselves against some other organisms. So are the plants are unique in this respect, or animals can do similar things?
I think animals can do have the same as plants in that they can contain lots of different chemicals that deter other predators from eating them, and little insects.
Yeah, definitely. But plants are much more inventive of producing a myriad of chemicals for all different purposes. So animals as well does it, so they produce different smells and pheromones and things like that. But the variety what plants can produce are millions. It's incredible store of new knowledge to dig deep into this chemical arsenal which can be done by plants. And there are posters as well around this topic, which I think we will come to that. So any further question? I see one from Enrique. So are cyanogenic compounds produced everywhere in the plant, or by flower or stamen or pistil? Where are they produced?
I'm not actually sure about that. I'd have to look it up, but I'm not sure if Daria knows anything.
I think we're assuming that they are in leaves and flowers. We don't know about the rest of the plant, but definitely in leaves and flowers.
I think it is a good bet that I don't know exactly either, but they would regulate where they are produced, this compound, and if it is for bee communication, it will be in flower. If it is used for other things, so they can regulate it. How they have specific promoters in these genes of the biochemical enzymes, which will put it to the leaf or to the flower or to the stamen or to the anthers to go exactly where it is needed. That's great. So if no more, then thank you so much again for all the talks, and please go to the Padlet and do your comments, and please go to the scoring sheet and give each other a voting that we can have this prize with a lot of votes backed up. So now we finish up with the last batch of posters. So the posters for this session starts with 15, which is coming from Jersey, and this Jersey poster is about telling the difference genetically and evolution sense between daffodils and the different daffodils. And this is actually a little, not a little, but an experiment which they carried out in this school, and they sequenced the chloroplast DNA of different daffodils they collected in Jersey in the spring. It must be very beautiful in that island in the spring. And the question is how related different daffodils are to each other. And they use a sequencing which is extremely fascinating, and again showing that that we are in a fast-moving train in biology, because this method was just discovered something like 5 years ago, how to sequence DNA with a handheld sequencing machine called nanopore sequencing. And what it does is actually let me go and show that that you can find, without me telling it, that you can find this how this all works, because we had a talk about it, and from the teacher of this group, John, presented a talk, and they also published it in a beautiful paper in Heredity as schoolwork. So please watch this talk as well together with this poster, and then you will learn how this nanopore sequencing is working. Okay, so that was about the daffodils and how they relate to each other. And the number 16 poster is No Species is an Island from Richmond School. And what this topic is about, in short, that organisms rely on each other in a mutualistic relationship, symbiosis relationship. And that is true between plants and microbes, or plants and nematodes, but it's also true between bacteria and ourselves, humans, and the human gut. And now, again, sequencing of microbes gives us the full picture of what sort of microbes are living in our gut, and what makes a healthy gut microbe population called microbiome, and and how our health dependent on these microbes. And such parameters like mental health can be dictated by the microbiome in our gut, but also other health factors like cancer, many other factors are connected. So this is a very active research. So this was nicely picked up by this group, and nicely presented. But many other aspects as well of this uh, dependence of the organisms on each other, also in Antarctica, for instance, and that talks about nematodes. Any comments on this one? I only see some campaigning going on for the vote. That's very good. So please vote for everybody, and choose yourself and listen to your instincts and your knowledge, which poster you like the best, or the talks. So the next poster is Monitoring Ecosystems Through Sound. And this is rhyming with one of our nice talk with Julia Kurocheva, which talked about how we can record sound in nature and actually make scientific conclusions by analyzing the sound. And the soundtrack will tell us biodiversity as well as how environment is changing by human activities. And this poster not only visually nice but nicely summarizing that topic. And if no questions, because I can't see, then we go to the next one. And that one again goes that plants are a factory of compounds because they have such a sophisticated so-called secondary metabolism, which is a small amount of metabolites produced by complicated enzyme pathways. And this was this is known for a long time, for instance, in ancient medicines. And this poster is taking us that by the knowledge we are developing, we can dig now deeper and deeper, identifying the compounds and synthesizing them. But because the synthesis of compounds are extremely difficult sometimes and also energy dependent. So now we are going more and more back to nature, and after we know the genes for the enzymes which makes these pathways, we can put them back to different organisms, either bacteria, yeast, or back to plants, and make so-called biofactories, which can produce a lot of goodies for our human well-being and human use. And this goes back to recombinant DNA technology as well, and genetically modified organisms for our use. So that was from Iona from Richmond School. Very nice. Picked it up. And the last one is again a nice post on a topical theme of the Pfizer vaccine, and this poster just nicely describes us what we need to know about this vaccine, and also how we are getting on with the different vaccines and immunizing the whole world. Hopefully, I hope you saw the the big summit we had, that the challenge is to gather the nations to have enough money that after we immunized our own country, we don't stay selfish, but we immunize the whole 10 billion of us in the world. So that's finishing the finishing the poster session. So any comments still? So if not, we will come to Ali with the last practical presentation. Ali, are you here?
Yep.
Okay.
Hi,
Hi. And after Ali's presentation, that you don't go away, I want really a couple of concluding marks so I really enjoyed this. So just to make sure that this conclusion is not disappearing, I will insert it before you, Ali. So I really enjoyed this masterclass, and we first time did it in an online form. So we do want to keep it up, and we rely on your feedback, and with the teachers we can keep the communication as well, how we should change and how we should run it next year. But I am really thankful for all the efforts both the teachers and the students put into it, and my colleagues, and it was not a burden at all. I really enjoyed running it. So I will come back with a little concluding mark, but I just wanted to say this, make sure that everybody hears. So Ali.
Okay. Thank you, Lassie. Yeah, nice to be here right at the end. It was really nice to see a bee poster towards the, well, a bee talk, sorry, towards the end there. For those of you that were there earlier in the series, so it was me who gave you a talk about bees in a changing world. And today I just wanted to finish off with giving you a flavour of how we run ecology practicals. And I'm just going to play you a video that I made earlier today about our fieldwork and how university level fieldwork works. I was also really excited to see in one of the posters there a description of using passive acoustic monitors, because that's what I'm going to describe. I'm going to show you how we use passive acoustic monitoring, so basically listening stations, to study rare bats. Latsi, I'm not sure if you, because the video's on the YouTube channel, I'm not sure if you were planning to play it or whether I should.
You can play it, Ali, if you can. So just share the sound as well.
Okay, let's I'll give it a try, but just stop me if it doesn't sound like it's working.
Okay.
Okay.
Or should I try it? Ah, you are doing it. Okay, that's great.
Yeah.
We see it. Yes, so let's see the
Okay,
sound.
okay, so let's check if there's sound. So hello everybody, I'm Ellie Leadbeater, and I want to, just in the last few minutes of today, give you a flavour of how we study ecology, field ecology, outside the lab when we go out into the field, into the real world. That's as large as it gets. And I'm going to talk about a field course that we've just that I've just come back from yesterday, actually, which was a UK-based field course. I'm really pleased that we are able to study ecology in the UK, because as we've all seen over the last 18 months or so, UK ecology can be absolutely fascinating, and it's incredibly diverse. You just need to know where to look. Where we went for this particular field course is somewhere called Knepp Castle Estate in Sussex. Knepp Castle is really particularly famous in the UK because it is our flagship rewilding project. And what people often think of when I talk about rewilding is introducing very exotic top predators. So, for example, wolves, lynx. These are all famous kind of rewilding ambitions. But in fact, the way rewilding works at Knepp is a far more holistic approach. To give you some background, Knepp was, until about the year 2000, was a working farm or a farm estate in Sussex. And things were going extremely badly, as they have been doing for many farmers. And that's because ever since the Second World War, we have been trying to eke whatever we can out of some very, out of very difficult to farm areas of land. So we've overused pesticides, we've tried to grow in extremely unfertile soil, which requires a lot of fertilizers. And basically it is becoming harder and harder to get that soil to produce anything. So the owners of Knepp, who were on the verge of bankruptcy at this time, decided to literally just give up trying to manage this land and try to get anything out of this land. And they just let things grow. They let things grow for 20 years. Now, they didn't entirely stop managing it. They took an ecological approach, which was to introduce large herbivores. You can see here we've got longhorn cattle and pigs here as well. And the reason for that is because they create diversity in the landscape. So we end up with a patchwork of clearings, forests, overgrown scrub, and what's really become quite a beautiful place. So the number of sightings of species that are not typically known in that area of the UK has been overwhelming. It has nightingales, it has nightjars, it has purple emperor butterflies, it has slow worms, it has storks. It is a pretty incredible place. I'm just going to let the owners of Knepp tell you about it in their own words, so that you can get a flavour of what this place really looks like.
conservation principles elsewhere.
The idea behind this project is now becoming well known as rewilding. The idea is you allow these natural processes as far as possible to happen.
Instead of conserving with specific species in mind, where you're focusing on keeping a habitat, locking it down as it is, so that that preserves the numbers of certain species. What we've done here is just taken our hands off the steering wheel and just stood back and let nature take over.
And of course, you then get wonderful things happening.
We've got nightingales, turtle doves.
Barbastel bat, probably Europe's rarest mammal, is starting to fly over this site.
In one cow pat alone, a student found 26 different species of beetle.
In a single morning, we counted last year 87 male purple emeralds, and that's exceptional for anywhere, really.
All 5 our species here.
A much more hands-off approach actually does deliver you the conservation benefits that you may predict, but also conservation benefits that you wouldn't have dreamed of.
What we're learning here, I think, is the future for conservation in the UK.
Okay, so that gives you a flavour of what NEP looks like and the kind of place that we go to do ecology fieldwork. Hang on. Without playing you that again. Ecology is not a luxury science. You can see we spend our time there camping, and that's so that we can get all our surveying done. We're up at dawn, we go to bed late, or up at dawn to hear the birds, and we're up late to listen to or survey the bats. And that's what I just want to give you a quick flavour of how we do it. So, bat surveying is a key skill for our ecologists to learn, and that's because bats, although they're not typically, then it wouldn't necessarily be the mammal that you might first think of when you asked about UK mammals. They are actually, we have, a quarter of all our mammal species are bats. Worldwide, there are over 1,400 species of bat. These are incredibly widely distributed. And in the UK, they are particularly vulnerable. And that's because they're pretty amazing creatures. So they they produce 1 offspring per year. They're incredibly long-lived. They don't even, well, most species don't reproduce until they're about 4 or 5. And female bats, when they produce young, they produce a pup, which is about a quarter of that bat size. And she has to carry that around with her for a week. So they invest, it costs a lot to raise a baby bat, basically. And that means that bats have become extremely vulnerable, especially when we develop the landscape. So keeping track of our rare species is a key skill that our students learn and that they need when they go out to their future careers. How do we survey bats? Well, I'm sure you guys know that bats hunt via echolocation. So they produce these really super high ultrasonic calls that bounce off moths and other insects that they're hunting for, and which the bats can use to effectively see the insects, although really they're seeing by listening. So the way that we analyse bat calls is we use bat detectors. What this is Vicky's showing you one of our listening devices. These are called audio moths. And what we do is we go out at night time, we put them in the trees, in the forests where we're interested in surveying the bats, and they listen for us. So they'll record all the sound that they hear. We set them to record for 1 minute in every 10. So throughout this forest, there's a horde of tiny listening devices to pick up the bats. I want to show you the kind of thing that we find using these. So let's leave here for a minute and look at one of our bat calls. So these are calls that we recorded last week, and I'm going to open one of our examples. Hang on. Let's look at this one. Okay, so what we've got here are 2 figures. Let me just zoom out. Along the bottom, we've got time. This is the beginning of the recording. This is the end of the recording. And up here, we have, this is what's called an oscillogram. So this tells us how much noise is going on at any any point. And you can see that in this recording, there is hardly anything going on at all. So we've basically recorded a minute of silence here. And this is what typically happens in any recording, because remember, these things are just set to record for 1 minute in every 10, irrespective of what's going on. So a recording like this, we can see there's almost nothing happening. So on this bottom spectrum, this is a frequency range. So high sounds would be up here, low sounds would be here. And this between 0 and 20 kilohertz is what human beings can typically hear. So this is the audible region down here. And up here, we've got the ultrasound. Now let's look through, see what we've got on our other recordings. So if we look here, we can see something much more interesting popping up. So remember, this is the audible spectrum down here, below 20, but these calls are very, very high. So these are without doubt bat calls. Let's zoom in on them and have a look at what they look like. Okay, so you can see they are very, very fast repeats of tiny chirps in the air. Let's zoom in a bit more. And these ones have a very kind of classic what's called a hockey stick shape. Okay, so I'll play you what they sound like. So this is a very, very slowed down version of a bat calling. So this is almost 1 second along here. You can hear that recording took much longer than 1 second. Basically, all these chirps are slowed down so that we can hear them, like slowing down an old style tape. You know, when you slow it down, it gets deeper. So that's exactly what's happening here. We're trying to make it deeper so that we can hear it. And in doing so, we have to slow it down. So what kind of bat is this? Well, a key feature in trying to work out what bat we're looking at, trying to identify species and see if it's a rare species, is the peak frequency. The peak frequency is the point at which the chirp is loudest. How high is it at that point? And that's actually the brightest point on the spectrum here. We're asking what frequency is that? And we luckily I don't have to look at this and kind of guess what frequency it's at. I can get the software to tell me. So I'm just going to highlight this bit. And we can see this tells us a huge amount of information about that particular call. But the key fact that I'm looking for here is it's telling me that the peak frequency is 56 kilohertz. Now, from that fact alone, in combination with the fact that their call looks like a hockey stick, I know that this is a soprano pipistrelle, because only soprano pipistrilles produce calls with a peak frequency of about 55 kilohertz. So there we go. We've identified what species of bat that was circling above us. And you can see if we go through the files a little bit more, on other recordings, we've got different bats. So here the peak frequency, I'm not going to look at it now, but it's certainly lower than 55. Okay, so we've got a huge range of bats that we found at NEP. Just to give you an idea of what some of them look like. Just to have a couple of examples. So here, this is a serotine bat that we recorded. Remember the peak frequency of the pipistrelle, the soprano pipistrelle was 55. If I look at the peak frequency for these calls, it's much lower. We're at 33 here. These are lower sounding calls. Have a quick listen to them. Again, hugely slowed down, but this tells us that this is a serotine bat. And at NEP last week, we found some incredibly rare species, including the barbicel bat, which is one of the rarest species in the UK. So that was pretty exciting for us. So that's given you an idea of how we look at bat calls. bats aren't the only thing we look at NEP. We do some other really cool stuff. So, for example, some of our students doing invertebrate surveys. They're looking particularly at the monarch butterflies, but other things that we find. We do moth trapping, find beautiful, crazy moths there, hawk moths, etc. This is a glowworm that we found last week on one of our surveys, and a great crested newt. So we learn about how to basically collect DNA from ponds and attempt to identify whether the great crested newt is present there. This is one of our most protected species in the UK. We also do things like mist netting of birds to track bird populations. So mist netting is, well, it looks unpleasant here, but it's actually very, very quick and a very low stress way to monitor bird populations. So you can see this is one of the finches that we caught. These are birds. This is a student releasing one of the birds back to the wild once the bird's been surveyed. There we go. Okay, and we also do reptile surveying. We find slow worms, we find grass snakes, we find adders, and finally we survey the mammals that are there. So NEP has a huge variety of large mammals, particularly longhorn cattle and the Tamworth pigs. But the deer, we had students doing deer projects looking at which landscapes the deer tend to aggregate in. Small mammal trapping. This poor little thing, it looks like it's being suffocated in the bag. I promise you it's not. We capture them very briefly in humane traps, and so that we can monitor the population size. This is a field vole. And the bats I've already told you about. So that's a summary of how ecologists go into the field, learn the skills that they need for future careers, whether that's in research or whether that's being a consultant ecologist, whether that's being a government ecologist, any of these careers which need you to be able to identify and understand animal population trends. Okay, so thank you for listening, everybody. I hope that gave you an idea of how we do ecology fieldwork and how we teach students the skills that they need to survive in the real world as ecologists. Thank you, Lazzy.
It's fantastic, Ellie. It's absolutely great. I am open mouth like they put a sign up in the chat box. So amazed what you are doing, and nice to see our colleagues in action. Thanks so much. You have some notes as well, Ali. Some people of your colleague nature, again, Jon Hale, I can read it out, that it's cool to see what data the Audiomos makes. Looks like that's the software probably used. And we set 3 up last summer. So this is the school in Jersey. And just send away the memory cards to analyze. I don't know, maybe. And another one was that, was it an eDNA protocol?
For the great crested newt, yes, that's exactly what it is. So sampling environmental DNA. So.
Okay, so just again a closing note is that it was very enjoyable, and the last bit that I will keep up the voting system for a day. Let's give it a day that you still can vote for the talks and the presentations, and I will correspond the results, and we will give out the prizes, and we will also we will send to the active participants who actually in the masterclass also presented either poster or the presentation, some documents that you participated in this masterclass. So that will take a bit of time, maybe 1-2 weeks, until the prices and this printed out documents will reach you. And thanks again, and good luck with everything, with your teacher assessed exams, and I know you are extremely busy, so thank you so much.
you